Hello and welcome, Doffy. I hope you're doing well. Thank you so much for passing by. Always great to see you. And yeah, I am back. I'm doing my best to stick to my schedule. And last week was a success. I should say the schedule that I posted in the Discord. And I'm also still working out sort of how to do appropriate graphics. It's all on a learning curve. But yeah, last week I had the goal of streaming 4 times per week, or rather 4 times that week. And I think I achieved that, at least from Sunday to Friday, or Sunday to Saturday. Regardless, like I posted a schedule in the Discord, and I stuck to it. And then on this past Saturday, I posted a new schedule on the Discord, and off to a rocky start since my intention was to stream on Saturday. That didn't pan out all that great. But, like, as mentioned before, my sleep is holding up. Still overall feeling good. But Saturday, I think what I ate on Friday wasn't really sitting well with me. And so I had planned stream that on Saturday or Sunday. But yeah, didn't end up working out. But I'm back. And I plan to make up for the lack of stream on, Sunday? Probably either a stream on Friday or Saturday. Still undecided. But again, moving forward for the next little bit, my bare minimum is trying to stream like 3 or 4 times per week, aiming for 4. And yeah, the schedule from week to week should be posted in the Discord. I'm also trying to stick to that. And again, still working out some kinks. But yeah, I think what I think is some great content prepared for today. Although hopefully I'll leave it up to the opinion of people watching. But I try to at least select papers that are interesting, but I usually just select papers that are interesting to me. And maybe not everyone shares the same interests as me. But I think just to hopefully make sure that I cover the topics that I want to cover. I specifically have like 2 papers that I want to cover. One more in-depthly, pretty much cover all of it. Try to break it down as best I can. It's a synthetic biology paper, and so it's definitely more in my wheelhouse, although my synthetic biology is a little bit rusty since I haven't really touched it much, like the past 5-6 years. I totally had a pretty decent or strong synthetic biology background, and like some first-hand experience with it since I've done some consulting, so to speak, for some synthetic biology researchers. But yeah, overall, want to do an in-depth look at that paper, and then sort of a less in-depth look at another paper about skin rejuvenation and exercise. Again, since the workout streams are on hold for now, so trying to cover health and fitness content, since that's also a passion of mine. Although maybe, again, not everyone is interested in the same things that I am. But I think it's a pretty cool paper, and I always try to at least share some of the benefits of exercise that is based in research. I think, finding out more and more all of the potential benefits of exercise and being physically active, but also at the same time, you don't need a high dose, or generally speaking, at least to gain some of the health benefits of exercise, you don't need to like beat yourself up or like exercise, 2 hours per day. Really, you can get away. I think I've covered research showing that even just like 5 minutes a day, or even just like small doses of exercise spread out over the course of the day, of like 5 to 10 minutes spread out over the course of the day, can lead to some pretty meaningful and significant benefits. I think we're just going to dive right into it to make sure that I cover everything that I want to cover. One moment. Okay. So there's this paper here that was published in the past couple weeks, or even less than that, literally last week, about, as it says in the title, engineered bacteria detect tumor DNA. And this is really like great synthetic biology, where generally speaking, at least the way I conceptualize synthetic biology, just pretty much biology and engineering just sort of smashed together, trying to, as highlighted in the title, engineer organisms, often bacteria, to do some desired function, and in this case, to detect tumor DNA. And I think it's really cool, not only just like what they do and kind of at some level its simplicity, but also the potential application of like, so this is still very much, as they say, in the experimental stage, not scaled up to humans. But if it actually gets to that point, which is not guaranteed, but if it does, like, there's quite a bit of potential. At least what they speak about holds quite a bit of potential. So, diving right into it, bacterial engineering has allowed the development of living cell diagnostics and therapeutics, including microbes that respond to gut inflammation, intestinal bleeding, pathogens, and hypoxic tumors. Bacteria can access the entire gastrointestinal tract to produce outputs measured in stool or urine. Cellular memory, such as bistable switches or genomic rearrangements, enables bacteria to store information over time. Some bacteria are naturally competent for transformation and can sample extracellular DNA directly from their environment. Natural competence is one mechanism of horizontal gene transfer, the exchange of genetic material between organisms outside vertical parent-offspring transmission. So yeah, understandably, not necessarily a concept that everyone or most people will be familiar with, but I think most people are familiar with how we as humans pass on our genetic information, as highlighted here, vertical from parent-offspring. So it's really, direct lineage from, the child that you give birth to, with, the shared genetic information of the parents. But then horizontal gene transfer, sort of as the term somewhat implies, it's not from, parent to offspring. It's pretty much like laterally, as in you can kind of think of it as like, siblings sharing DNA, or like, two bacteria next to each other, just like exchanging genetic information. But really it's not through a descendant or offspring. It's just pretty much an exchange of genetic information. It would be kind of, as a very loose analogy, me just like, giving some of my genetic information to like, a friend of mine, if I had the ability to do that. And there are circumstances in which bacteria can essentially uptake DNA from the environment, as they're pretty much going to exploit here, or we can kind of like force DNA inside bacterial cells, which we often do in order to engineer them, and they are engineering bacteria here. So, as they highlight, horizontal gene transfer, or HGT, is common between microbes. It may also occur from microbes into animals and plants. So that's also pretty much just about everything else outside of a vertical gene transfer, as in that it's not parent-offspring. Pretty much, more or less, in the horizontal gene transfer domain. But there is a little bit of an exception there, as they're kind of gonna, as I highlighted here. So yeah, it may also occur from microbes into animals and plants, and in the opposite direction, from eukaryotes to prokaryotes. And we are eukaryotic organisms, and prokaryotes are generally speaking, bacteria. So it's like, it can be from eukaryotes to prokaryotes, but going in the opposite direction, not necessarily the case. So yeah, the forward engineering of bacteria to detect and respond to mammalian DNA through horizontal gene transfer, however, has not been reported. So yeah, you can have from eukaryotes to prokaryotes, but prokaryotes to eukaryotes. That's pretty much what they're trying to attempt here. Acinetobacter bailey is a highly competent and well-studied bacterium that is largely non-pathogenic in healthy humans. Can colonize the murine gastrointestinal tract and acquires unpurified environmental DNA from lysed cells. So this is essentially, part of our microbiome, or can be part of our, native microbiome, and pretty much doesn't cause any disease or condition, at least as they say, in healthy humans. there might be some circumstances in which maybe like an immunocompromised human can, let's say, have some negative effects from the bacteria. But they're obviously choosing this for a reason, as in, like, if they want to actually scale this up to humans, and like, yeah, they ideally want something that is not pathogenic or that won't cause a problem to the host. So yeah, it can colonize the murine gastrointestinal tract and acquires unpurified environmental DNA from lysed cells. Our cellular assay for targeted CRISPR discriminated horizontal gene transfer, or CATCH as they say, strategy delivers bacterial biosensors to sample and genomically integrate target DNA in Figure 1. So this is their overall schematic here. They have their engineered sensor bacteria. So engineered ALE bacteria are delivered rectally in an orthotopic mouse model of colorectal cancer. The naturally competent ALE take up tumor DNA fed into the colorectal lumen. The tumor DNA is engineered with a CAN-R cassette flanked by CRAS, which I'm going to elaborate on further, but this is essentially a gene for antibiotic resistance, specifically the antibiotic kanamycin, and they're using this for a specific reason we're going to get into. So, oh yeah, and I should probably sort of go over the figure, at least the left side of the figure, in a bit more detail. Like, yeah, so in their like schematic or in the figure, you have your colorectal cancer, and they are a source of genetic information. And you have your engineered bacterial sensors. And so if through horizontal gene transfer, they uptake some of the genetic information of the cancer cells or tumor cells, well, then a genetic event will happen. That's more or less what they're depicting here in the top right of the diagram. As in, sort of like the blue line, you have your cancer cell donor DNA, and then you have the bottom brown line, you have bacterial sensor DNA. An exchange of genetic information will happen without dissecting, the exact mechanism. But yeah, there's essentially genetic exchange happening, or that is pretty much the horizontal gene transfer. And if the genetic exchange happens, and they will pretty much they will gain the function of kanamycin resistance, as in they will acquire the function of being resistant to that antibiotic, kanamycin. And then if they grow them in the presence of that antibiotic, then they should grow because they now have the resistance, as opposed to pretty much that is their signal, as in if there's the presence of cancer, then they will be able to grow in the presence of the antibiotic, as opposed to if there's no cancer, when the antibiotic is present, well, they will die because they won't have the antibiotic resistance. It's essentially a binary. So, to demonstrate this concept, we use the biosensor to detect engineered tumor cells. We then developed genetic circuits to detect natural, non-engineered tumor DNA sequences, discriminating oncogenic mutations at the single base level. Because the target sequence and output gene are modular, our approach can be generalized to detect arbitrary DNA sequences and respond in a programmable manner. So what they're highlighting here is this is a proof of concept, a little bit, where it's like, yeah, if this works, then it should not, in theory, be isolated to just this. As they say, it's modular, and it should be adaptable, again, in theory, to a number of other purposes. Probably not just isolated to colorectal cancer. And so, let's see. Dissecting the figure more, pretty much going over what they say about the figure. Yeah, engineered Bailey bacteria are delivered rectally in an orthotopic mouse model of colorectal cancer. The naturally competent Bailey take up tumor DNA shed into the colorectal lumen. The tumor donor DNA is engineered with a kanamycin resistance cassette, flanked by CRAS homology arms. The sensor bacteria are engineered with matching cross homology arms that promote homologous combination. This is pretty much how they allow for this horizontal gene transfer, as in the cancer cell or the tumorous cell, giving some of its genetic information to the engineered bacteria. So sensor bacteria that undergo horizontal gene transfer from tumor DNA acquire kanamycin resistance, that's that antibiotic, and are quantified from luminal contents by serodilution on antibiotic selection plates. So it's like, yeah, basically as a result of the cancer or the cancer cells donating some of that genetic information through horizontal gene transfer, they gain this new function, as in resistance to the specific antibiotic, kanamycin, and then they should be able to grow in the presence of that antibiotic. That's essentially, well, that is what they're depicting here at the bottom right, where this is pretty much just your standard agar plate with, you know, nutrients, and then you have the antibiotic there throughout the entire plate. And like, yeah, if there's cancer, they've acquired now this resistance gene, well, then they will be able to grow in the presence of that antibiotic. But if there's no cancer, then they don't acquire resistance, and then they will die. Pretty much the signal is, if you have cancer, you get bacterial growth. So, engineering cancer cell lines, organoids, and sensor bacteria, here's are the results. To test the hypothesis that bacteria could detect specific mammalian DNA, generated transgenic donor human cancer cells with a kanamycin resistance gene inside Cras homology arms. Going back to that figure. Cras is an important oncogene, the gene that promotes cancer in human cancer, and a driver mutation in Cras often accompanies the progression of simple into advanced colorectal adenomas. Our technology is currently confined to the detection of specific sequences, and thus for cancer detection is limited to hotspot mutations, as Cras G12D.
Show me the money.
Oh wow! Thank you so much for the sub, the peer Greg from the peer review. Super appreciated. Thank you so much for passing by. Also super appreciated. Hope you're doing well. We stably transduced. 1 second. Yeah, microbio, obviously best bio. I can't disagree there at all. Although if you're including microbio and in the overall, let's say, if you're including molecular in that as well, or if you just mean micro, because is this like strict microbiology? I don't know. But I don't really want to get into the dividing lines of micro versus molecular versus synthetic versus biochemistry versus chemical biology, etc., etc. Yeah, our technology is currently confined to the detection of... Oh, I already read that. We stably transduce this donor cassette into 3... Yeah, exactly. I know there's some very loose dividing lines between, you know, molecular and, let's say, biochemistry. But, like, yeah, at some point they all get, like, really mixed and matched in my brain. And actually, fun fact, so I'm in the department of chemistry and biochemistry, and my PI is like a strict, well, fairly strict biochemist. That's their overall training, but I think virtually all of the grad students that he has had, or like 90% of them, have come from the biology department, or like they're all pretty much like molecular biology. So, you know, I think he has some success with them so far. I might be the outlier. We stably transduce this donor cassette into 3 conventional human colorectal cancer cell lines. So they're starting off with a cell line, a human cell line. So again, they haven't scaled up to humans. That's not in the scope of this paper. But they pretty much, they they do work up to sort of organoid, well, mice organoid. But I don't think in fully, well, in the full animal mouse model, I think it's mostly just like, you know, organs from mice. Oh yeah, especially like this, well, just going back to the beginning, like obviously research like this is just like a huge interdisciplinary endeavor. And it's like researchers from all over the world collaborating, pretty much just going through all the affiliations here, from like LA to Australia. Those seem to be like the major areas where it's like, yeah. And I overall like understand it, although some of my synthetic biology is rusty, and even some of my genetic knowledge is also rusty. But even then, like I can't dissect every single thing as much as I would like to. But I do like going over it, like super cool. Yeah, I got like this is just like a huge interface between like so many disciplines. And that's why I probably like just as it probably the reason why it requires like so many disciplines just to like understand the entirety of this research. So yeah, we stably transduced this donor cassette into 3 conventional human colorectal cancer cell lines with differing background genetic alterations. Here are sort of the genetic alterations of the cancer cell lines, or the colorectal cancer cell lines. So, and they did it using a lentiviral vector. This is sort of standard genetic, or pretty standard genetic manipulation in human cell lines. Aren't gut migrants kind of local to parts of the GI tract? Well, so I do believe that we have pretty much like bacteria throughout the entirety of our GI tract. But like, yeah, I think the major concentrations, or like the highest density, I guess you would say, are in like the large intestine. But I do believe we have pretty much like there's a microbiome like throughout an entire gastrointestinal tract, but I think it's just like way more limited. We're always like in the stomach, not the most favorable conditions for bacterial growth, but I still think you have bacteria growing there. Or at the very least, you know, you have the infectious H. pylori growing in your stomach. So yeah, I don't think this particular strain of bacteria is ubiquitous. But it could be totally wrong about that. I'm an E. coli person, so I have too narrowly focused on E. coli. That pretty much anything that's not E. coli is pretty foreign to me. Yeah, exactly. That's my understanding as well. But I think the focus is like just generally speaking on like the large intestine, since like I think that's largely driven by the fact that it's like the most accessible. And I think my knowledge could be a little bit outdated because I haven't, you know, maybe followed up as much as I should have. But like, you know, a lot of like the microbiome research are like trying to like assess what's in our microbiome, like comes from like pretty much like analyzing, you know, pretty much what we output in our stool. But even then, there appears to be like if I remember correctly from when I did some digging into research, of like there's a little bit of a disconnect. Of like, yeah, just because, well, what you output is not necessarily fully representative of like what's in your actual microbiome. And there's obviously there's understandable sampling issues with, you know, getting up into, you know, the small intestine, or like pretty much trying to sample every portion of your GI tract. But I'm sure there's technologies being developed that like in pill form, you'll probably be able to like swallow some sort of pill that will take samples as it passes through your GI tract. I know we do that with, let's say, capsule endoscopy, of like taking photos of your GI tract as you swallow a capsule. Well, I imagine that at some point there'll be the capsule that you can just swallow and it'll just sample as you go, I think. Oh, April, welcome. Thank you so much for passing by. I hope you're doing well. And I hope you're also feeling well. Matters the most. To construct the sensor, and right now we're going over this paper here, engineered bacteria to detect tumor DNA. Yeah, like I only know of the capsule. I'm most familiar with capsule, and I say that because capsule endoscopy is a thing. This is sort of one of the more modern ways of trying to, do a colonoscopy, but like through most of your GI tract. But obviously there's the drawback of it's just photos. Where very often for your standard colonoscopy, they do want to like actually take biopsies, and be able to like, see at the cellular level, as opposed to like this is just images. But I'm sure the technology will be there at some point, to like start sampling. Probably. Ooh, I'm sorry to hear that. I hope that it was in. I know that wisdom teeth removal, from what I understand, can range from, at least I only share my experience of me getting my wisdom teeth removed was like fairly straightforward and like a 45-minute procedure to get all four done, because they were all pretty much whatever, fully formed. They didn't need to like dig them out, or really be extracted. They just kind of need to like yank them out. And so I had like very minimal swelling. I had just like some mild pain for like 24 hours. I would say arguably just like mild discomfort. And then after 24 hours, I was pretty much like fine. Well, except for, obviously, the holes in my gums. But I know for some people, like they have pretty awful experiences with wisdom teeth removal. And they get, lots of swelling, and if they have to be like extracted, there's obviously like way more pain, and discomfort. So like I fully recognize I got pretty lucky. My wisdom teeth. Yeah, that's exactly the challenge. Where for very often, capsules sort of like very often used as like preliminary, of like, hey, maybe let's just like take a look and see if we need to follow up and go the more invasive route of a colonoscopy. But I think the technology of capsule endoscopy is getting like more and more better over time. Well, just like every other technology. But like, 15, 20 years ago, like not a thing at all. And I think it's way more, at least from what I understand, it's way more popular in the United States, where I don't know of anybody really, in Canada, at least in my vicinity, that has undergone a capsule endoscopy. But one of the other nice parts about it is like you just like swallow the pill and they give you kind of like, whatever, a little backpack, that you can just like go home. And then you're more or less like, having the procedure done as you go about your day. And like, you're just like saving, in the hard drive in like the backpack. So that's pretty cool. Oh, I'm super sorry to hear that. I'm hoping you can get some rest and recovery. One was impacted. Yeah, I know that's a very common experience for many people that have had their wisdom teeth removed. I know that I got very lucky with my wisdom teeth. Like it was much quicker than I anticipated. But like, yeah, I hope that you can pretty much get back to normal, and get back to feeling well as soon as possible. Hopefully, it's just a matter of time. And also, I'm assuming that getting your wisdom teeth removed is for the long-term benefit. But hopefully, you have now an excuse to like whatever, eat a bunch of ice cream, and food, and foods that you hopefully enjoy, that don't cause you issues. That was one of the quote-unquote downsides for me, is like I didn't really have much of an excuse to eat a bunch of like bad food. because like, yeah, I was more or less back to normal in like 24, 48 hours. Rafa, welcome to ALS. I used to watch a streamer who had ALS, and he talked about gut bacteria a lot. Are you referring to Lou Gehrig's disease, as in amyotrophic lateral sclerosis? So I don't. So I'm definitely familiar with the condition. You know, everyone at least knows of, let's say, Stephen Hawking, and they had that. Although I think, if I remember correctly, Stephen Hawking was a little bit of an outlier in terms of like how long they were able to live. But yeah, I actually don't know the relation between ALS and the microbiome. I'm actually now curious, because from what I understand, ALS is largely genetically driven. But that doesn't mean that it can't be influenced by your environment. I mean, just like with virtually any condition, that even if it's like fully genetically driven, let's say cystic fibrosis, that's also heavily influenced by your environment. As in, like, very often individuals with cystic fibrosis, they are dealing with chronic lung infections as a result of their genetic condition. But I might have to do a little bit of research. I'm not going to do full research now. I'm just going to do a quick. See if there are any articles. Do. Well, so this is just like me reading one paragraph. So obviously, you know, I'm still largely ignorant. I'm just like 0.1% less ignorant. But all they're saying here so far, but this is just like the one paragraph I read, which is not surprising, in that individuals with ALS, amyotrophic, lateral sclerosis, their microbiome is different than healthy controls. Like that makes total sense. Just intuitively. I'll probably have to save this paper later. Maybe go over it on stream. But like, yeah, I mean, the gut microbiome, like, the research there is, like, as far as I understand, in its infancy, although it's exploding pretty heavily. But we're just like sort of like finding out more and more how interlinked, or you know, how the gut microbiome affects so many things, presumably. But we're still trying to tease out. So even using my own condition as an example, you know, I have generally speaking, you know, a severe GI condition. So there's quite a bit of research on, you know, the microbiome and Crohn's disease. But even then, the relationship is still a little bit unclear, or at least it's not fully clear, in that there appears to be some sort of effect of the microbiome, but we don't know exactly where, you know, what they say here with amyotrophic lateral sclerosis. They're saying, hey, when we look at patients or people with ALS, their gut microbiome is different than people without ALS or healthy controls. I mean, that makes perfect sense, in that people with ALS, you know, your gut microbiome is very often influenced, if not largely influenced, by your diet and what you eat. And so it's like, well, almost certainly individuals with ALS, they will likely have a different diet than healthy controls. Probably like a very specialized diet or something, probably more specific to them. And so that will almost certainly alter the microbiome. And the same thing holds true with, you know, again, patients or individuals with my own condition. Like Crohn's disease patients, they typically have, let's say, fairly unique diets, or at least diets that are different than, you know, your average healthy control. And so when we look at the microbiome, the gut microbiome of Crohn's disease patients, like we find differences. We find like very clear differences. Generally speaking, we find much less diversity. And so many people, understandably, but falsely link the two in terms of like, oh, since their gut microbiome is altered, then that must be what is driving the condition, as in driving the inflammation in Crohn's disease. And it's not that simple. Or at least it's not as simple as that, because, again, your gut microbiome is influenced not only by what you eat, but it's influenced by, the local environment. All that to say is like the changes that we see in gut microbiome of Crohn's disease patients, we don't fully yet know whether that's the disease or a result of the disease. We don't know cause and effect, as in it could be as a result of the inflammation and the diet that Crohn's disease patients have, that they just happen to have a different microbiome than healthy people. Or is it that, yeah, the gut microbiome changes somehow, and then that results in the disease? There's been some emerging research recently, I think in the past like 1 or 2 years, showing that like they're not so cut and dry, where there appears to be some influence of the gut microbiome in terms of like, it seems like it can either maybe drive the inflammation or make it worse. But we're still not sure, or at least it's definitely not concluded with any certainty that, hey, it's the gut microbiome that is causing it. Because there's totally research showing that, like, once you treat them, do find it. Yes. So, many people understandably think that, again, if you look at Crohn's disease patients, their gut microbiome will be different. And so it's like, well, then it's obvious that whatever, it's the gut microbiome that is driving the condition, or it's like causing the disease. But then there's research that I'm showing here, where it's like, if you actually, if you treat the inflammation, okay, and then you go and reassess the microbiome, like, okay, we've treated the inflammation, or at least, dramatically reduced the inflammation, and we've like changed local environment, then like now what does the microbiome look like? And it tends to look like, or tends to pretty much move in the direction of a healthy control, as in they pretty much like start looking like a healthy control. Where, as they sort of say here, treatment with ADA induces short-term changes in the microbiota composition, which seem to parallel the partial recovery of the gut bacterial ecology, with recovery parameters tending to eubiosis recovery. This is more or less just, fancy terminology for, whatever, healthy controls. So it doesn't appear to be so clear-cut, and this research is not, super old, in 2015. But there's emerging research to show that, like, yeah, there is some role there for the microbiome, and even there in terms of like maybe driving or some sort of role in the disease process, but we're still not quite clear what that is. That does appear to be true from my understanding, April. But even then, at the end of the day, yeah, the research on the microbiome, there's still a lot of unanswered questions. And like, I don't know for sure, but sort of last time that I looked into the research, like, we don't know, like, what the optimal microbiome is. So far, from what I can tell, all we're doing is like, we're sampling, quote unquote, healthy people or healthy controls without defining what that is. And we're saying, well, these people don't have, any major health conditions, so that must be the healthy microbiome. But like, is that actually true? And there's this notion put out there, of like, well, generally speaking, the greater diversity that you have, well, the healthier your microbiome is. But there's some research to show that, like, yeah, decreasing your diversity, or it's not necessarily true that, you know, low diversity in your microbiome is like inherently bad. Yeah, there is some research to show that, like, well, actually through decreasing or reducing the diversity, like this person or group of patients had positive benefits. So again, still a lot of unanswered questions, but the research is exciting, for sure. And it's research that I try to track for multiple reasons. One, because it's interesting, and two is because, like, yeah, I have a gastrointestinal disease, so and there is some sort of role in the microbiome. I've had small intestinal bacterial overgrowth for 6 months now, and it's not going anywhere with the natural treatments. And that, yeah, that is also, I think, an issue that we're grappling with, of, like, you know, reassessing you know, our antibiotic usage. Where I think as a result of this research, among, you know, other things happening in the world in terms of antibiotic resistance, but, like, yeah, it seems like we, you know, let's say 15-20 years ago of, like, just, hey, for pretty much like whatever infection, just like carpet bomb with antibiotics. Maybe we need to reassess that approach and be more measured and targeted with our antibiotic use. Because, like, yeah, unfortunately for people like yourself, April, it's like, you know, it can really do a number on your gastrointestinal tract, and can sometimes take a long time to recover, or, like, don't ever fully recover. Or maybe as a result of, like, antibiotic usage, maybe like some sort of you know, micro change that results in a gastrointestinal ailment. Where I think more and more some research is also coming out, not surprisingly, that obviously microbiome and IBS, irritable bowel syndrome, etc., etc. So yeah, lots of, like, overlapping stuff. Oh, you know, to apologize for derailing me. I'm almost live for derailment. How does your brain keep track of all the information you read? Do you ever find yourself? Totally. So I try to keep track, and it might look that I'm keeping track of everything, like, but it totally happens. For sure. Like, I have thousands of papers, and the thousands of papers that I have, they range from ones that I've read from beginning to end, from, like, you know, from the title to, like, the end of the discussion, and then there's papers there that, like, I've just, like, browsed through, and then there's papers there where I've just, like, read the abstract, and it totally happens where it's like, you know, I have something in my mind of, like, you know, I'm going through my filing cabinet in my brain of, like, I have this piece of information in my brain, and it's linked to a paper, but I don't remember you know, what paper that came from. And it totally happens where I'm just, like, searching. Thankfully, this software has a search function, because, like, yeah, I would be, like, very be super challenging without it. Where, like, yeah, I can spit out information, but I always want to at least provide some reference for what I'm saying. And it totally happens where it's like, yeah, the information is there. I can't remember from which specific paper, or, like, I definitely wouldn't be able to remember the title, but it's like some keywords. It's like, oh, that's literally what I did here. When I brought up how when you treat Crohn's disease patients, their microbiome tends to, like, normalize or move in the direction of, you know, healthy controls. So I had that piece of information in my brain, and all I did is, like, well, obviously I have some organization of, like, well, I have my folder of Crohn's disease research, and I happen to remember what drug they were using for that study, and that's a little bit of luck on my part, but I knew that it was, like, adalimumab, so ADA, and then microbiome, and so I literally searched ADA space micro, and thankfully it showed up. Where if I didn't have those two keywords, I'd be totally lost. There was no way that I would remember, like let's say this title. So it's definitely not, perfectly organized. Mostly just like I have a lot of information gathered in there, but I totally rely on like software like this and search functions, while you retrieve original reference. So all that to say, it's a lot of show, smoke and mirrors. And yeah, don't apologize for derailing. If there are questions, and I can answer those questions, pretty much always willing to answer. But getting back to this, where so they are constructing their bacterial sensor, and so we left off here. What do they do to construct their bacterial sensor? So they inserted a complementary landing pad with chromology arms into that bacterium, a Bailey. We tested both a large insert design, where 2 kilobytes of donor cassette must transfer, and a small insert design where only 8 base pairs must transfer to repair 2 stop codons. I'm gonna, dissect this and provide hopefully more clarity in a bit. The initial biosensor output was growth on kanamycin. So yeah, sort of harkening back to this figure here, where again, their overall workflow is like, yeah, they have their bacterial sensor, or sensor bacteria that have been genetically engineered to be a certain way. And then, they have their colorectal cancer model, and it's a specific type of colorectal cancer. And that's what they go through here, of like the genetic alterations, where it's not just like any run-of-the-mill colorectal cancer. It's like these specific types. But this is not to say that, probably their overall long-term ambition is like, yeah, to hopefully maybe detect not just any colorectal cancer, but maybe like any cancer or a number of different cancers in different parts of the body. But, regardless, they're starting off, a focused way. And, more of a simple way. Simplified model. But yeah, they have their engineered cancer cells. And then some of these cancer cells, they will shed or, output genetic information. And some of that genetic information will be taken up by these engineered sensor bacteria. And through the exchange of genetic information, as they say here, cancer cell donor DNA, as in the cancer cell is giving some of the genetic information to the bacterial sensor. So it is an exchange of genetic information. And their signal is if there is an exchange of genetic information from the cancer cell donor, then the engineered sensor bacterial, they'll be given the new function of being resistant to the antibiotic kanamycin. And that's why they have this antibiotic selection plate, as in if there is the presence of cancer, then that exchange of genetic information will happen, and the bacterial sensors will acquire that antibiotic resistance, and then they will grow in the presence of that antibiotic. But then, if there's no cancer, they don't get the resistance, they don't gain that new function, antibiotic resistance, so they will die. They are put on the growth plate. And then, so here, sort of dissecting a little bit closer, what they're actually doing at sort of like the genetic level, that I will dissect in a bit. So we tested these designs using various donor DNA sources, both in liquid culture and on solid agar. Figure 2A, this figure here. The large insert biosensors detected donor DNA from purified plasmids and genomic DNA both in liquid and on agar. This is, agar and liquid. The major difference is, liquid is liquid. Agar is pretty much like that solid plate that's highlighted here in the bottom right. Your standard kind of like Petri plate that contains a solid medium, or pretty much just like, a solid gel-like substance that provides nutrients for the bacteria to grow. Or just for the bacteria to be there, but they may die because there's also an antibiotic there, and they may or may not have resistance, depending on the conditions. On agar, they also detected raw, unpurified lysate, albeit at just above the limit of detection. Okay, so we're going to dissect the figure a bit, just go over all of the results. As expected, the small insert design improved detection efficiency tenfold, reliably detecting even raw lysate. Across conditions, detection on solid agar was more efficient than in liquid culture. Notably, these experiments confirmed that the biosensors did not require DNA purification. And then lastly, before dissecting the figure, mutations in codon 12 of CRAS occur in 27% of colorectal cancer tumors, accounting for 72% of all colorectal cancer CRAS mutations, and are common in solid tumors generally. To test whether sensor bacteria could discriminate between wild type and mutant CRAS, used A. Bailey's endogenous type, blah. Yes, I'm gonna return to that. Okay, so we're gonna go sort of somewhat systematically in the figure. Starting in the top left in the A section. So yeah, donor DNA was derived from plasmid purified cancer cell genomic DNA or raw lysate top that recombined into biosensor A. Bailey cells. Bottom, horizontal gene transfer included either a large 12 kilobyte insert or a small 8 base pair insert repaired to stop codon. Although that's not what I would say, that's not a super critical detail. No. But what they're testing here is how much sample processing needs to take place in order for their system to work, right? Because again, their overall, their ultimate goal, somewhere down the road, 1, 5, 10 years, 15 years down the road, is like maybe we can use this in humans to detect colorectal cancer, okay? And, their ideal scenario, as, highlighted here by essentially, scientific version of essentially a poop emoji, is like, they want to be able to, again, in humans, just from sampling, their waste output, their stool sample, well, can you just answer the question of whether there's colorectal cancer just from that? Or rather, like, from that with, like, very minimal sample processing. What they're testing here is, like, various methods of sample processing. Or it's like, how much sample processing does it need for the bacterial sensor to work, as in acquire that genetic information? Because again, the genetic information, ideally, somewhat ideally, is coming from a cancer cell that's, enclosed in something, and it contains genetic information. So somehow, the genetic information, in order for there to be exchange of genetic information between the cancer cell and the bacterial sensor that they've engineered, well, pretty much, the genetic information is going back to this figure. The cancer cell donor DNA needs to come into close proximity to the bacterial sensor DNA, as in that exchange of genetic information, like, has to pretty much, it's an event, has to happen, and, there's pretty much, they need to be essentially in close proximity. So, what they're doing here is, well, can the genetic information come from, the cancer cell, or do we need to pretty much, like, extract the cancer cell, and then, extract the DNA from the cancer cell, and, purify the DNA, and then, inject it into the bacterial sensor. And then the genetic exchange happens, and then our bacterial sensor, gives us the output, as in, that growth in the presence of the antibiotic. Or, can we, does it need just like straight, pure genetic information? In the form of what, a plasmid, without dissecting what it is, that's just basically circular DNA. So they're testing essentially various forms of DNA purity. From like the most pure, this is just like pure circular DNA, and like, pretty much like delivering it into the bacterial sensor, the biosensor. This is sort of like the most processing required, as in this is essentially the less ideal scenario. They don't want to have to, like, take the stool sample from human and then, purify all the genetic material and then like give it to the bacterial sensor. And then, yeah, there's various forms of like, so starting with the cancer cell donor, they will purify the genetic information from there. That's what this little tube represents. And then, it's delivered to the bacterial sensor. Or can they just kind of like burst open a cancer cell? There's pretty much like, there's virtually no purification steps. It's just like, hey, we've burst open the cancer cell, and then we just like deliver, that soupy mixture of, burst open cancer cell that contains, all of the contents of the cancer cell in addition to the genetic information, and we just like deliver that, the biosensor, and then, the genetic exchange will happen. That represents essentially like minimal sample processing, as we don't really need to do much manipulation. We could probably just do that kind of like straight from like maybe a stool sample, or like, maybe minimal sample processing of a stool sample, as opposed to like, purifying some of the genetic information from a stool sample. That's what they're essentially testing here. When they say, yeah, donor DNA was derived from plasmid, so that's like, pretty much ultra pure DNA. And then purified cancer cell genomic DNA, so that's, one step more pure, or rather less pure. And then the raw lysate, that's pretty much, crude, messy, contaminated sample, but it does contain the genetic information that we want. And then, like, yeah, it recombines. And what they're showing here in B is the different types of, let's say, so when there's exchange of genetic information, they can essentially exchange a lot of genetic information, or they can exchange a little. And that's more or less what the 2 KB, hopefully the cursor, but that's what they're referring to here. Find it. Nope. Yeah, it's a large insert versus the small insert. Fine. Well, they're essentially like testing somewhat the limits, as in, can we exchange basically, a lot of genetic information, or like a little, and how successful or not with either strategy. But the overall principle is like still the same. They're just like testing the extent of genetic information from the cancer cell that is being delivered to the biosensor. But yeah, in both cases, conferring kanamycin resistance, D to G. Oh, they're referring to this here in the middle, the graphs. So yeah, in both scenarios, whether it's, a lot of genetic information that's exchanged or a little genetic information that's exchanged, the output is the same. They will both get, well, in both scenarios, they get resistance to that antibiotic, kanamycin. And then, so that's D to G. And then... Yes. Oh, okay. Okay. Gonna get to that. So, we tested these designs using various donor DNA sources, both in liquid culture and on solid agar. The large insert biosensors detected donor DNA from purified plasmids and genomic DNA, both in liquid and on agar. On agar, they also detected raw, unpurified lysate, albeit at just above the... I think I read this. But I'll continue reading it. As expected, the small insert design improved. Yeah, blah, blah, blah. Yeah, well, we did read that. So the sensor bacteria should reject wild-type CRAS through CRISPR-mediated DNA degradation, but allow integration of the CRAS G12D sequence. Two of the three spacers block transformation by both wild-type and mutant DNA, figure 2I, and yeah, I'm going to break this down in a bit. However, spacer 2, for which the CRAS G12D mutation eliminated CAM site selectively permitted only CRAS DNA. The other common mutations in codon 12 of CRAS all eliminate CAM as well. Thus, sensor A Bailey can be engineered to detect a mutational hotspot in the CRAS gene with single base specificity. Right. So, like I mentioned, figures B and C. So, yes, they tested, let's say, whether there can be an exchange of a lot of genetic information and, be successful, or an exchange of a little genetic information, and can it still be successful? The answer is yes. Both scenarios, whether it's a lot of genetic information or a little bit, then both outcomes are successful. But then moving on from there, they wanted to test sort of the resolution of their system, as in, okay, can we actually detect like a very specific form of cancer, or a very specific mutation of a cancer, because cancer, very broad term, and even just like colorectal cancer. Definitely not an expert on colorectal cancer, or pretty much any cancer for that matter, but I know enough to say that even there are a lot of genetic changes that occur on any cancer. Even just like narrowing in on colorectal cancer, not every colorectal cancer is the same. Where pretty much there are certain genetic changes that occur with or certain specific genetic changes or mutations that occur with some colorectal cancers, and then there are other specific genetic mutations that occur with other colorectal cancers. All that to say is that just as cancer is heterogeneous, colorectal cancer is also heterogeneous. And so they're pretty much asking themselves the question of like, hey, there's essentially this specific type of mutation in this type of colorectal cancer, and we detect it. So instead of just asking, the broader question of like, hey, can we just like broadly detect colorectal cancer? They're going from the broad to the specific of like, hey, there's this mutation that's associated with this type of colorectal cancer, and we detect it. So that's what they're referring to here when they say this. Mutations in codon 12 of CRAS occur in 27% of colorectal cancer tumors. This specific mutation occurs in a proportion of colorectal cancer tumors, and they account for 72% of all colorectal cancer CRAS mutations. These are a specific type of mutations, and are common in solid tumors generally. So to test whether sensor bacteria could discriminate between wild type, this is just a fancy way of saying, the non-cancerous, or pretty much like the non-mutant type, the one found in the healthy population at large, the non-disease type, and mutant crass, but they're talking about a specific type of mutation. It's called crass 12D, so it's pretty much a very specific mutation. We used A. Bailey's endogenous type IF CRISPR-Cas system. We stably transduced a donor RKO cell line with the CAN-RGFP, green fluorescent protein, donor cassette flanked by wild-type Cras, and a second line with Cras P12D flanking sequences. Next, we designed 3 CRISPR spacers targeting the wild-type Cras sequence at the location of the Cras12 mutation. I'm going to break this down in a bit. Using the A. Bailey protospacer adjacent motif, PAM, we inserted these as single spacer arrays into a neutral locus in the large insert A. Bailey sensor G. This is the number one. Totally, it's understandable, since if I remember correctly, at least when it comes to cancer lethality, I think colorectal cancer does have one of the higher fatality rates. But I think a lot of that, again, if I remember correctly, is driven by pretty much not enough screening and detection. Where, yeah, it's one of the more lethal ones, but with appropriate screening and detection, it can very often it can be like prevented or caught very early. I think that's one of the major hurdles when it comes to colorectal cancer, unfortunately. And I'm super sorry to hear that. My heart goes out to you. And I'm totally, I myself am totally at risk, much higher risk of colorectal cancer, given my condition. And I am followed pretty closely. Definitely closer than your average other person that's in my age demographic. I think generally speaking, I think it's like, I think the recommendations are like, if you're over the age of like 50 or like 55, it's recommended that you start being screened for colorectal cancer. Well, through colonoscopy, as in you start having regular colonoscopy. That's already been occurring for me since I was like 11 years old. And the longer and longer that I have my condition, just like my risk is like just gets higher and higher. I mean, it's not super sky high. It's not like a guaranteed, but totally, I am like a pretty substantially elevated risk. But it's a little bit of there's the positive side of, well, since they take regular looks through, with a colonoscopy, since I'm subjected to regular colonoscopies, there's a very high likelihood that if I were to get it, it would be caught early, or like, if there's a potential cancerous polyp, it would be removed, and the issue would stop there. So gotta take the good with the bad. Screens are being moved from 5 to 3 years. Yeah. I don't, even though I've literally had probably maybe approaching 100 at this point, or maybe close to triple digit, maybe dozens and dozens. But like, yeah, they never get easier. That's sure. And I'm pretty good with like, the preparation and the procedure. I definitely obviously don't enjoy it, but now it's sort of like routine to me, of like, I'm just like kind of like numb to it. I definitely don't enjoy it. But like, yeah, it's, that's why I bring up capsule endoscopy. Hopefully we get to the point that like just a capsule can like serve like, or can be an appropriate substitute for like, the invasive procedure. It is 50 years of age once every 10 years. I believe so. I think that was maybe a recommendation at one point. And obviously I'm in Canada, so it might be a little bit different in terms of like guidelines. But I think there's, I don't know if it actually has happened, but I think at one point there was a call to like revise the guidelines. At least like every 10 years is like too long. And it should be like every 3 to 5 years or something to that effect. Maybe I should update. Yeah, so yeah, even then they've reduced the age at starting. Most people should begin screening after turning 45, then continue at regular intervals, but they don't say the years. But like, yeah, as obviously more often than other people, if you have number 1, inflammatory bowel disease such as Crohn's disease are also correct. Obviously fit that category. How often? Yeah, they don't say how often, but yeah, I think in addition to like lowering, like I think this 45 is a somewhat recent change, and I think they leave it either has happened or like they've reduced it, or there's calls to reduce the intervals in which you get screened. A proper diet. So take what I say definitely with a grain of salt. But from what I understand, although I am pretty confident in my knowledge, but at the end of the day, I'm definitely not a medical professional. But from what I understand from the research that I have read, like diet, generally speaking, diet higher in fiber does affect your risk. As in, I think it's generally recommended that if you want to further reduce your chances, incorporating pretty much your standard healthy diet recommendations pretty much fall in line with reducing your risk of colorectal cancer, as in fruits, vegetables, higher in fiber, etc., etc. Hey, I don't blame anybody for complaining. I don't blame anybody for complaining about their first, obviously not, or like their second or their 10th. Like I said, I'm on my like whatever dozens and dozens, and I still complain a little bit. I mostly complain about obviously like the preparation and like the fasting and like clearing out your system, even though like I have that since I've been through it so many times, like I have it down, largely to a science of like, hey, I know more or less like when to start fasting, or like the week leading up to it. I use this diet and cut out whatever XYZ from my diet, and then it makes the clearing your system out much easier for me. But even then, it's still largely a not fun experience. Nervous to be put. Totally, that's also totally understandable. Hopefully I didn't fearmonger the other day about intraoperative awareness or conscious awareness during general anesthesia. But I'm curious, like, if you want to share, are you nervous? Are you nervous about going to sleep or not going to sleep, or like the effects of sedation, or something else, or an allergy? Went to a capsule feeding right now. My endo only said that she won, but if she saw something questionable, yeah, that's exactly it. Where for someone like me, it's pretty much like a cost saving measure, as in like At the end of the day, they want to be able to do biopsies, you know, in my gastrointestinal tract. They can pretty much, uh, they can only get limited information from just visualizing from a capsule. And even then, if let's say there was like a visual anomaly, they would pretty much similar to your situation, they would want to like go in anyways, and take biopsies. I was like, well, might as well just like go straight to the invasive procedure. Like pretty much, unless they're like highly confident that everything that they see will look healthy, there's pretty much like no reason to do a capsule endoscopy on me. And there's no reason to think that my GI tract will ever look healthy visually. These are reduced by 5 years though, if I recall. Yeah, exactly. I think that's one of the reasons why they reduced it. Like, yeah, 45. It's because I think they recognized that not enough people were being caught early. I can say without a doubt that my mother was a tiny woman, but her diet wasn't good. She was simple and busy, have the same lunch forever, a glass of Kool-Aid, a small bag of chips, and cheese, processed meat every day. Yeah, and it's unfortunate, but like diet is tricky, right? And I don't judge anybody for having a suboptimal diet or not having the perfect diet, because the perfect diet doesn't exist. And even though like I try to follow a healthy diet, there are definitely days or weeks where I don't have a perfect diet. And like if your mom was, like you said, they were busy, and like when you're busy and you're on you're in that go mode, which I have fallen into in the past, of like, hey, I'm just too busy to make conscious choices about my diet, so I'll just like go with what's convenient. And no surprise, what's convenient unfortunately doesn't always turn out to be the most healthy, or more often than not doesn't turn out to be healthy. And so it's like, yeah, I'm super sorry to hear that, but it's an understandable situation. And I've totally been in that situation myself. And like, yeah, I'm super sorry that your mom passed away. And that, yeah, my heart goes out to you. I wish I had better words than that. It's like, that's just like a difficult situation. Vagina problems that are not diagnosed. Oh, CFC, thank you so much for the kind words. I appreciate it. And also, thank you so much for passing by. Super appreciated. And like, yeah, if you find my content awesome, if anyone finds my content awesome or informative, that's really all I ask for, and that's pretty much why I'm here. And this what encourages me to keep going. Adrenal problems that are not diagnosed, a ton of allergies, MTHFR, and slow COMT variation, plus all the unknown, I'm afraid. Okay, yeah. Then I totally understand. For sure. I think though that's regardless of anyone's health status, being concerned about sedation or anesthesia is a totally valid concern. And like, given what you just shared, totally super understandable and valid concern. As someone who's been under just about every type of sedation or anesthesia under the sun, if I were in your position, I would be super paranoid as well. Oh wow, thank you so much. That's super kind, and I'm super grateful for that. And that's why, like, I think this is going over this because I think it's super cool, and I think this is hopefully what's in the pipeline. Maybe not in the immediate pipeline of like next year or even 5 years. This is probably like 10, 15 years down the road. But I think this holds, as we'll see when we get towards the end, this holds a lot of promise, potentially. Still in the experimental phase. But yeah, what they moved on to is like trying to detect like a very specific type of cancer, or a very specific mutation of, a variety of colorectal cancer. And so they're doing they're using a very specific strategy that, I don't necessarily want to dissect, but I can sort of try to give, the broad overview. So people, I think virtually everyone now these days has heard of CRISPR, and just, they know it as a gene editing technology. And CRISPR is essentially, we have kind of like hijacked it, for our own gene editing purposes, but it exists in nature. Like it's coming from bacteria. You can essentially think of this as the bacterial immune system or defense system. That's pretty much what they're saying here. We used a Bailey's endogenous specific type of CRISPR-Cas system. Endogenous, just fancy word for essentially, internal or native, as in contrasted with exogenous from the outside. It's a Bailey's, internal CRISPR-Cas system. It's what they have, native to them. And this is part of their, think of it as their immune/defense system. As in, bacteria can get infected. They can get infected by phages. They're pretty much infected by viruses. And so just as we humans have our defenses and, immune system to combat, pathogens, both bacterial and viral. Bacteria themselves, they also have their own defense system. And that is that CRISPR-Cas. What they're doing here, okay, is, the CRISPR-Cas system that is native to this bacteria, or to bacteria at large. Its overall purpose is to degrade DNA, but to degrade specific types of DNA, because they, you don't want to degrade your own DNA, as in the bacteria contain DNA, and they don't want their own CRISPR system to destroy themselves. So it's a targeted, it is a targeted DNA degrading system. And so what they're doing here, okay, that's what they're highlighting here in the bottom left in this figure, H, is they've designed, they're sort of hijacking the A. Bailey's, the bacteria's CRISPR-Cas system, their immune system, and they're using what they call spacers, okay? And these spacers might be difficult to see, and this is where I'm going to invest in a tablet soon, just so I can start like drawing and zooming in closely on things. But all that to say is like they are providing specific DNA sequences to their engineered bacterial sensor, okay? And the principle is, okay, their ultimate end goal is like they want to be able to detect a specific mutation, okay? Like one single mutation, and that's highlighted in this box here. So it's referring to H. So how do they describe H? CRISPR spacers targeting the CRASP G12D mutation box. So that's a single base pair mutation. That little box there. So it's really just what we call a point mutation. Not just it's not some large change in genetic information. It's really pretty much it's the smallest unit of genetic change that you can have. Just like one single base pair. Literally one letter change. So yeah, they're targeting that CRASP G12D mutation, that box, using this underlined PAMs. Fraction of total biosensors expressing the indicated CRISPRs were transformed by plasmid donor DNA or plastic. Okay. Yeah. So, principle is, they only want to detect, a very specific sequence. A sequence that contains just that single base pair change. So what they need to ensure, or what they're trying to ensure, it's like, hey, if there's anything else that's essentially similar, okay, or rather it's similar but not identical to this specific sequence that they're looking for, it will be degraded. or rather, it should be degraded. That's the principle of like, hey, this this DNA sequence, yeah, it's similar to the type of cancer that we're looking for, but it's not identical. But since it's not identical, we've designed it such that it should be degraded by that CRISPR, the bacteria's immune system, that they are hijacking. That's why they say that they are hijacking it. Where do I probably missed it? Yeah, endogenous. So the principle is, the bacteria's immune system should degrade everything else, okay, except for the DNA sequence that contains that one single base pair change. Pretty much like they just want that to go through alone, as in to bypass the bacteria's immune system. Everything else should be degraded. And that's essentially what they're kind of like testing out here, of like with these spacers, of like, hey, this is essentially close, but not identical. Was it going to be degraded? That's what they're hopefully going to answer. But like, hey, this genetic sequence, it's exactly what we're looking for. And so does it get past bacterial defenses? And whatever, spoiler alert, as we're going to go through, it does bypass the bacterial defense. As in their system works. As in pretty much it's able to detect that specific genetic sequence that contains their mutation of interest. So that's what they highlight here. The sensor bacteria should reject wild-type CRASPs through CRISPR-mediated DNA degradation, but allow integration of the CRASP G12D sequence. So again, they're only looking for a specific sequence, and everything else should be rejected by being degraded by the bacteria's immune system, CRISPR-Cas system. So 2 of the 3 spacers blocked transformation by both wild-type and mutant DNA. So they are testing a mutant. They're testing, let's say the wild type. That's, think of it as, healthy DNA, or like, what's contained in the healthy population at large. And then like, yeah, they are testing a mutant DNA, but it's not you can have various different types of mutation, but it's not the specific mutation that they're looking for. So they're testing, regular run-of-the-mill DNA sequence, and then they're testing, a different mutant genetic sequence, and then like their third one is like, hey, this is a mutant, but it's this specific sequence. So that's why they say 2 of the 3 spacers block transfer, as in pretty much the system worked. The only one that got passed through bacterial defenses, like that exact genetic sequence that they were looking for, that contained their mutation of interest, and that this other mutation that they were not interested in did not make it past the bacterial defense system. However, Spacer 2, for which the CRASP G12D mutation eliminated the PAM site, selectively permitted only CRASP G12D donor DNA. The other common mutations of both in codon 12 all eliminate this PAM as well. Thus, sensor A Bailey can be engineered to detect a mutational hotspot in the CRASP gene with single base specificity. They can pretty much like just a proof of principle of like on paper, it looks like their system is high resolution. Like it only allowed a very specific DNA sequence through to be incorporated into the bacterial sensor, and then the bacterial sensor to do its thing of like gaining antibiotic resistance, and then growing in the presence of the antibiotic, as in success, essentially. So, detection of cell-free DNA, tumorigenic organoid lines. So, up until this point, they've been working with engineered, or like genetically engineered, not only genetically engineered bacteria, genetically engineered human cell lines, or cancerous human cell lines. But that is essentially, or it is in vitro. But again, the ultimate end goal, but like very ultimate end goal, is like in humans, fully human organisms. So they're scaling up a bit. Obviously, like I said, in this paper, they're not going to be testing on humans, but now they're going from human cell lines to organoid lines. So it's pretty much just like a little bit of a step above of like, hey, can we get closer, one step in the direction of a fully formed organism instead of just like cells in a dish. So we evaluate our sensor and donor constructs in organoid culture. We previously used CRISPR-Cas9 genome engineering to generate blah 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 mouse organoids. They're pretty much they are saying here how they made their organoids. So we transduced BTRZI organoids with the donor DNA construct to generate donor colorectal cancer organoids and incubated their lysate with the more efficient small insert A. Bailey biosensors. And this is going so just as when they were working with cells in a dish, okay, human cancer, colorectal cancer cells in a dish, these were genetically engineered in a specific way. When they're using now organoids, they are also engineering them. So these aren't just like run-of-the-mill organoids. They are very specific to like what they want. Okay, this is the type of kind of like colorectal cancer in organoid form that we want to like analyze and assess for our system. And then they are also using their previous results in this next set of experiments, as in what they highlighted here, going back, is like, yeah, in their original system, in just cells in a dish, they tested whether their system can have genetic exchange with a large amount of genetic information or a small amount. And what they found is that the small amount, both were successful. But it seems like the small amount, the 8 base pair, what they call the small insert, that worked more efficiently. Both worked, but this one pretty much just like worked better. So given that fact, they're going with the small insert design for their next round of experiments in the organoids. And that's why they say, yeah, small insert. Because like, yeah, more efficient. We use quantitative polymerase chain reaction, qPCR. We confirmed that the BTRZ organoids that we generate contain only 2 copies of the target donor DNA. As with the CRC cell lines, the sensor A. Bailey incorporated DNA from donor organoid lysate, but not from control lysates from the parental organoids. Next week, co-culture GFP. I'm going to break this down in a moment. The parental or donor organoids are 24 hours in matrix gel. The GFP expressing sensor bacteria envelop the organoids after co-culture with the donor, but not parental organ. A. Bailey sensor bacteria acquired donor DNA through horizontal gene transfer. Finally, we estimated the detection limit for biosensor for target DNA in stool. To achieve this, we added increased amounts of donor plasma to define. Okay, yes. So, all that to say is like they largely repeated what they did when they were working with cells in a dish, but now they're working with organoids. But there are some slight differences. So, because again, an organoid system. So, schema depicting in vitro co-culture of A. Bailey sensor bacteria with the BTRZ CRASP-CanR, the colorectal cancer donor organoid lysates or viable organoids test, horizontal gene transfer repair of kanamycin resistance gene. So, starting here in the top left, panel A. So, what they're assessing here, okay, is overall the same strategy that they started with here in this panel A. Of like, okay, They tested how much sample processing needs to be done when they were working with just the cancer cells in a dish. The overall same principle of like, hey, how much sample processing do we need to do when we're working with organoids in a dish? And so here there is some minimal processing, as in they took some of the organoids, they broke them open to expose the genetic information, and then they assessed whether their bacterial sensor would, essentially function or have that genetic exchange event, and then, work. But then they also tested, oh, Moon, welcome. Thank you so much for passing by. Good to see you. I hope you're doing well. Right now, we're covering this paper, engineered bacteria to detect for DNA. Not this one, sorry. So, yeah. They start off with the colorectal cancer organoid that they, designed and engineered. And then they're testing, well, does it need some processing for our system to work? Or can we just go straight from like, organoid in a dish also, or like, in like a sample tray. And that we don't really need to do any processing. Just the bacterial sensor will be present in the organoid. And then from there, it will essentially be able to do its thing. As if we pretty much need to do like a very minimal amount or like nothing to the samples. Pretty much like don't need to process the samples. And it seems like, okay, that's what they're going over here, is in B and D. So B is the lysate, as in they, just as going back to this panel, they took some of the cancer cells and they broke them open to expose all of the stuff, including the genetic information, and then observed whether the system worked, and it does. They're doing the same thing here, of like taking some of the organoids, breaking them open, releasing all of the stuff, including the genetic information, and then seeing if the bacterial sensor works. But then they're also doing the one where, hey, we're not going to break anything open. We're pretty much going to do minimal sample processing, and then see pretty much like virtually no sample processing, and then see if our system works. And it looks like it does. It works maybe a little bit less efficiently, but it still works. And that's more or less the comparison between B and D, where B, lysate is just fancy terminology, or, technical biology terminology for like, hey, breaking cells or, breaking biological material open to release all of their contents. We lyse them, we break them open. That is actually what the term lyse means. Which is like, yeah, just breaking down of the membrane of a cell. So, yeah, they break open the organoid to release a bunch of stuff. And again, they're testing, well, can we get away with not breaking them open? And it looks like they can. Where if you compare panels B and D, so on the left-hand side of the graph, well, if you look at the x-axis, you have pretty much the control, okay, no signal. Well, a negative control, that is what you would expect with a negative control. And then their donor, like, is pretty much the comparison, or like, their system working in comparison to control. And then with the organoid co-culture, that pretty much you get the overall same results. The number is a little bit reduced, but it's really not all that significant, as in both system works, and it looks like they can get away with basically not breaking the organoid cultures open. And here their signal is a little bit different, because going back to their preliminary, or like their first round of experiments, they were starting with cells in a dish, and then their signal that their system was working, or rather their signal that they are detecting cancer, as in, hey, there's the presence of cancerous cells here. The indication is bacterial growth, as in growth in the presence of that antibiotic. They're getting the antibiotic resistance from the cancer cell's genetic information, or that exchange of genetic information with the cancer cell donor. But here, there's still an exchange of genetic information, but their signal is no longer growth in the presence of an antibiotic. It's pretty much, well, it's green fluorescent protein. It's pretty much it's a visual signal. Where, let's see, we're using quantitative polymerase chain reaction. I mean, it's highlighted here in panel C. That's what that, that green sort of halo is. Like, hey, pretty much, their signal is like glowing or fluorescing, to be more technical. Yeah, recombination with DNA from crude lysates enables growth of A. Bailey sensor on kanamycin. Representative images of GFP tagged A. Bailey biosensors surrounding, yes. Oh, okay, no, I made a mistake. Sorry, no. They're still going with that kanamycin system, as in, like, yeah. If there's the presence of cancer in this organoid system, like, yeah, it's still antibiotic resistant, and they will grow in the presence of the antibiotic. But they do move on to GFP at some point. Let's see. Gonna move on. Gonna return to this figure to dissect it more. Where was that? Yes. Welcome, boy. First of all, welcome, Boy Meets Mini. Nice to see you. You're back. And hey, I'm sure that's in the pipeline. And also, shout out to you. I hope you're doing well. Nice to see you. If you're not following Boy Meets Mini, definitely should be. Yeah, I feel you on the tired part. Sleep is like a chronic issue for me. But getting slowly better. Next, we evaluate our sensor and donor constructs in organoid called... Yes, I already read that, I think. Yeah. Sorry. Given that cancer to bacterial horizontal gene transfer occurred in vitro and in the presence of stool, we sought to test the catch system in vivo. We first confirmed that our BTRZ orthotopic colorectal cancer model released tumoral DNA into the colorectal lumen. Engineered colorectal cancer organoids were injected orthotopically by mouse colonoscopy into the mouse colon to form colonic tumors, as previously described. Using digital droplet PCR, we measured BRAF mutant tumor DNA in stools collected from tumor-bearing and control mice. The BTRZ model reliably released tumor DNA into the colorectal lumen. We next conducted an orthotopic colorectal cancer experiment. Immunodeficient okay, well, I don't think it's critical. And she mice were injected with donor or non-donor organoids, or neither. At week 5, once the tumors had grown into the lumen, sensor or parental A. Bailey bacteria would deliver it twice through rectal enema. The mice were subsequently euthanized, and the colorectum harvested with the luminal effluent plated for analysis. So dilutions were then plated on agar with different antibiotics. Okay. So yeah. So they're moving on. So they're describing the lower panel of figure 3. That's an excellent question. So, hypothetically, like on paper, it should be pretty early. Because you shouldn't need like a lot. So, their system is like detecting um cancerous genetic information, as in you have, some growth of cancerous cells, in this case, in the colon, because they're using the model of colorectal cancer, but it, on paper, it should be adaptable to other cancers. Um, but all that to say, uh, shouldn't need the presence of a lot of cancer cells for this system to work. They should just need a little bit of genetic information uh, for their system to work. Where, like, yeah, uh, they sort of test limits of detection, and let's just say it should be almost virtually way almost guaranteed to be virtually way earlier than something like, let's say, conventional screening methods of, let's say, with colonoscopy, like that. Because, typically, in screening for colorectal cancer through colonoscopy, like it's like through visual inspection of your large intestine, as in, like, hey, if we either like see a polyp or like something precancerous, well, whatever, we either like remove the polyp, or like we do a biopsy, as opposed to you only need you don't need like visual amounts of the cells there in this system, or you shouldn't need, in theory, visual amounts. Like, you should just need like microscopic, like, this is pretty much like it would be before it would be before you get to the point of, like, you can actually see this, the colonoscopy. That is one of the primary advantages. And as they will get to, there's another key advantage of, like, this hypothetically should not only be useful for detection or screening, it should also be useful potentially for treatment as well, or like a 2-in-1, both a detection and treatment, because, again, this is all proof of principle of, like, yeah, all of the components here. On paper, they should be exchangeable with other components and used for different things. That I'm sort of going to attempt to break down once we get towards the end. But hypothetically, on paper, you should be able to couple, like, again, if this system holds true and it can be useful for humans, then you should also be able to use this for a treatment as well, or even a combination of both detection and treatment. Visual is definitely too late. The more, yeah. Although, even so, yeah, definitely, earlier the better. That is for certain, for pretty much just about any disease. Where was you're right. So here at the bottom, so they're pretty much going in a fairly systematic way. As we saw at the beginning, they started with cells in a dish. Pretty much a pure in vitro experiment of cancer cells in a dish, engineered colorectal cancer cells in a dish, and then they went up to organoids. Okay, so pseudo in vivo, or like in between in vitro and in vivo, maybe in situ, whatever. Not all that critical for the terminology, but all that to say, the organoid system worked. As in, their system is still working once they've scaled up to a little bit more complicated system. Now they're trying to do, or attempting to do, full in vivo, as in full mice, or rather like hybrid mice. Unfortunately, probably no surprise to anybody. We are experimenting on mice here, and we're kind of like doing uh, not so not the most humane things per se. But like we are like manipulating them, and grafting things to them. In this case, to see if the system still works, scaling up from pretty much full-fledged mouse model. But yeah, given that cancer to bacterial horizontal gene transfer occurred in vitro and in the presence of stool, we sought to test the catch system in vivo. So all that to say is like their cells in a dish system worked, that in vitro. Now they want to move into in vivo, and that is what they're calling their system, their engineered bacterial sensor. It's the catch system. So we first confirmed that our BTRZ orthotopic colorectal cancer model released tumoral DNA into the colon lumen. Engineered colorectal cancer organoids were injected orthotopically by mouse colonoscopy into the mouse colon to form colonic tumor. Oh wow. Thank you so much for the raid, and welcome, raiders. Super appreciated. I don't know what to say. But yeah, I am Corey Campbell. I'm a biochemist. I cover science, research, and news. And right now we're going over this piece of research here about engineered bacteria to detect cancer or tumor DNA. And welcome, Wildcat Snow. I am doing well. And Tyler, hope you had a wonderful stream. And shout out to you. If you're not following Tyler, you should be. And I believe I was lurking a little bit in your stream. I think you were testing your thetan levels, if I remember correctly. I have no doubt that your thetan levels were off the charts. Thank you so much for the follow, my weird uncle. I also appreciate the username. But yeah, I hope you had a wonderful stream, which I have no doubt that you did. And I can't thank you enough for the raid. Super generous and super appreciated. And welcome, everybody. I am Corey Campbell. We're going over some research here. We're pretty much, we're engineering bacteria to see if we can detect cancer. But we're using an animal model. And they first started with cells in a dish. And their bacterial system worked. Cells in a dish. Cancer cells in a dish. And then they moved on into organoids. Okay, a step above cells in a dish, and it still works. Now what they're doing is they're generating essentially like hybrid mice or engineered mice to see if they can detect colorectal cancer in mice, or specifically, human colorectal cancer in mice. And yeah, they are manipulating these mice in, maybe not the nicest of ways. But yeah, they performed a mouse colonoscopy. And they pretty much gave them colonic tumors. I was like, yeah, they want to see if their system works in mice. So that's what they have to do. And then using digital, we measured BRAF mutant tumor DNA and stools. Yes. So once they, induced cancer, human colorectal cancer, in these mice, they wanted to see if, like, okay, if the cancer is there, and is it actually shedding genetic information? As in, like, yeah, our intention was like to give these mice, colorectal cancer, human colorectal cancer, and they're pretty much like confirming, were we actually successful in that? And well, yeah, the answer is yes. They confirmed, like, hey, we tried to give them cancer, and it looks like we did. And then now they want to see if their system like works. So we next conducted an orthotopic CRC experiment. Immunodeficient NSG mice were injected with donor or non-donor organoids, or neither, at week 5, once the tumors had grown into the lumen sensor. So, yeah, again, they're giving them cancer in the form of organoids. They're allowing them to grow and to actually, like, force develop into like full-fledged cancers. And then they're going to see if their engineered bacterial sensor like actually works. As in, can our engineered bacterial sensor actually detect this cancer that we've given to the mice? And that's what they're saying here. As in that sensor, the bacterial sensor, the engineered bacteria were delivered twice through a rectal enema. So they are giving these mice, enemas. The mice were subsequently euthanized and the colorectum harvested with luminal, well, pretty much sample processing. Where it's like, yeah, they gave the mice cancer, and then they are giving them their engineered bacteria to see if they can actually detect cancer. Horizontal gene transfer from tumors to biosensors was only detected in donor tumor-bearing mice that were administered sensor bacteria. There was no horizontal gene transfer detected in any control group. All that to say is that their system works. In that they only detected cancer when pretty much all the appropriate things were present, or like the necessary things present. And that's actually what they're showing here in this graph F. Of like rectal delivery of their engineered bacterial biosensor to mice bearing colorectal cancer donor tumors results in kanamycin-resistant A. Bailey biosensor in luminal contents through horizontal gene transfer, blah, blah, blah. So they're testing like how efficient was like that exchange of genetic information, and it turns out it was pretty efficient. And so here in panel F that I've just described, you have here pretty much, on the far right of the graph, that's pretty much like their signal of like only when there was the donor tumor DNA, that positive sign, and their sensor, as in they got the sensor, did they actually pretty much get a signal of like they got what they wanted. When anything else was absent, or any combination, or any different combination, the system did not work. As in, if they only got the sensor, but there was no donor tumor, well then the system didn't work. But then if they got the sensor, and then yes, there was a donor, but it wasn't a tumor, or it's a non-donor tumor, then the system still didn't work. Pretty much it's only when you got exactly what they wanted did the system work, as in a sensor specifically detected the donor tumor. So where was it? Yes. The resistant colonies were confirmed to be the engineered biosensor strain by antibiotic resistance, green fluorescence, and genetic sequencing. Right, because again, this might be a native bacteria to the mice. So, as mentioned in the beginning, in the introduction, like this specific type of bacteria that they're using is pretty much often found in healthy humans, or maybe also in healthy mice. And so they want to make sure that, they are not detecting, native bacteria, or like the native strain of bacteria that's already there. They're trying to distinguish it between like the actual engineered bacteria that they've introduced. And that's why they sequence them. And that's what this 16S sequence is alluding to. Of like, hey, we've, recovered the contents, or, we've essentially dissected the mice and processed them, and then we're going to see what bacteria are there. And then we're going to make sure that it's, our specific bacteria that we've engineered are actually present and there by sequencing the genome of the bacteria that are present. And horizontal gene transfer mediated can repair of individual colonies, thus hatch, which that's their system of engineered biosensor, discriminated mice with and without colorectal cancer in our experimental model. So, as in their biosensor behaved as intended, as in it detected cancer in the presence of cancer, and only in the presence of colorectal cancer. Finally, we designed living biosensors to detect and analyze non-engineered cancer DNA. Right. So, up until this point, they have been looking at, they are looking at colorectal cancer, but they are looking at very specific, types or varieties of colorectal cancer. Because colorectal cancer, just like cancer is a broad term, also colorectal cancer is a broad term, as in it can manifest itself in, almost an infinite number of ways. But their experiments up until now, it's like, hey, we're only going to look at this specific type, or this specific genetic background of colorectal cancer. But the system might have limitations if, like, hey, it can only detect this type of colorectal cancer. They're proving the principle that, hey, it can detect this type or this type. But what about just like virtually any colorectal cancer? That's what they're moving on to. And that's why they say living biosensors to detect and analyze non-engineered cancer DNA, as in sort of like more run-of-the-mill. Because, when someone gets cancer, such as, let's say, in myself, if I were to get cancer, it would be in a non-engineered way of, like, pretty much whatever bad luck or whatever bad lifestyle choices or combination thereof. So they're pretty much saying, asking, how broad is our system? Yes, it can detect this specific, but its ultimate utility is how broad it can be. So, the TET-R repressor gene was inserted between the Cras homology arms in the biosensor, and in a second locus, we placed an output gene under control of the blah 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 promoter. Here, the output gene was canamized in resistance for ease of measurement, but the output gene is arbitrary and exchangeable. Right. So I'm going to break this down in a moment, but it's a slightly different system. Their ultimate end signal is the same, as in that that antibiotic resistance, but like they say, it doesn't have to be this. It doesn't have to be, resistance to this specific antibiotic. It could be a different antibiotic, or it could be a totally different signal altogether. As they're sort of saying, their system is modular. They're just going with something simple, and that's, widely used, as in, in biology, giving bacteria antibiotic resistance is like pretty much every run-of-the-mill biology lab ability to do that, or any run-of-the-mill biology lab that works with bacteria has the capability to do that. This is like essentially standard practice. So in this design, expression of the output gene is constitutively repressed upon recombination with the Cras target DNA. The repressor TET-R is deleted from the genome if the incoming Cras sequence is wild type at the G12 locus, cascade type IF CRISPR Cas effector complex detects and degrades it. However, if the G12 locus is mutated, AMSite and therefore the CRISPR-Cas targeting are eliminated, and expression for the output gene turns on. I'm gonna break this down. Do I do it now? Yeah, I'll do it now. So, up until now, they've essentially been working with a somewhat simple system, although definitely not downplaying what they're doing here, because overall what they're doing here is like very cool, complicated, and as highlighted earlier, it requires like a whole team, a whole multidisciplinary team of experts and researchers around the world. But their overall kind of like proof of principle system is fairly simplified. That pretty much there's they're engineering bacteria that has the ability to exchange genetic information with a cancer cell, a colorectal cancer cell. And if there's that exchange of genetic information, well, then the engineered bacteria, they gain a new function, and that new function is antibiotic resistance, specifically resistance to the antibiotic kanamycin. Here, they're switching it up a little bit. It's still kanamycin resistance, but it's a little bit more of a complicated that I'm going to attempt to break down. So TET-R, located between the homology arms on the A Bailey genome, represses expression of the output gene. Target DNA with wild-type CRASP sequence is recognized and degraded by the type IF CRISPR-Cas vector cascade. Target DNA with the CRASP avoids degradation, replaces TET-R in the expression of the output gene, DNA fraction of biosensor, target donor DNA. Okay. Yes. Here in their final set of experiments, as mentioned, their ultimate goal is they want to, as they say, detect non-engineered cancer DNA. So what they need to ensure, it's like, hey, only a genetic sequence that is cancerous gets detected. Whereas before, generally speaking, it's only specific sequence. It's engineered cancer that's detected. But now they got to make sure that they essentially that they don't get false positives, as in the biosensor is only sensing what they want, and in this case, it's just broadly colorectal cancer. So what they need to ensure is pretty much everything else that they're not interested in gets rejected. And we got hints of that earlier when they were using the CRISPR-Cas system with this. When they were looking to test whether their system has the ability to just detect, one single point mutation, and it worked. And their strategy was pretty much these other sequences will be degraded by the bacterial defense CRISPR-Cas system. So they're using that again here, where pretty much everything else should be degraded, but what should be accepted, and that will allow their system to work. But what happens is appropriately B and C. So target DNA with wild-type CRAS is recognized and degraded by the target DNA CRAS avoids degradation. R and resuppression, yes. Okay. Thinking. Yeah. Right. So, in the absence of nothing, okay, that's A here. So this TET-R, that is also an antibiotic resistance, but it's for tetracycline, just a different antibiotic. The other one was kanamycin, and it was called CAN-R. This one's TET-R, so it's tetracycline resistance. But all that to say, is their system is that pretty much in the absence of nothing, okay, their output here, okay, that's pretty much the green arrow. That's their output. But in the absence of nothing, okay, their output is basically shut down. That's what the line with the blunt head represents. That represents repression. So all that to say is their output when there's pretty much nothing present, pretty much no genetic information, either from a non-cancerous or cancerous cell, pretty much the default state is like their output is repressed. But if it comes into contact with genetic information, but in this instance, in panel B, the genetic information is coming from wild type, as in essentially wild type CRASP. That's essentially, that should be non-cancerous with genetic information, but it's non-cancerous genetic information. So what should happen, this cascade, that's essentially part of the CRISPR-Cas system, and so that should be degraded, and that's why they say the target DNA with wild-type CRASP. So this is not what they're looking for. They want this to be degraded, and they're saying it should be recognized and degraded by that CRISPR-Cas effector complex. They just call this this particular part of the CRISPR-Cas system cascade. But their actual target DNA with their non-engineered mutation avoids degradation, replaces the tetrapressor in the biosensor genome, and relieves repression of the output gene. So it's pretty much the opposite of panel A, where when it's actually in the presence of pretty much, non-engineered cancer, then this pretty much essentially removes this TET-R that acts as a repressor to their output. So if you remove the repressor, well, then their output is no longer repressed, as in it's pretty much switched from the default off position to the on position. You're pretty much removing the off signal from this TET-R system. That's pretty much the function that it serves. It's the TET repressor. Pretty much primary function is to repress. So yeah, the presence of non-engineered cancer or genetic information that's coming from non-engineered cancer cells, that off signal should be removed, and then their output is turned on, or like the absence of the off signal automatically turn on their output. So in this design, expression of the output gene is constitutively repressed, as pretty much this A panel here. Like I said, pretty much their on signal, their output is constantly turned off, and that's why they reference figure 4a. Upon recombination with the CRAS target DNA, the repressor TET-R is deleted from the genome. If the incoming CRAS sequence is wild type at the G12 locus, cascade, the type IF CRISPR CASP effector complex detects and degrades it, and that's, figure 4b. So, if it's like, again, not cancerous, wild type, then pretty much that will be degraded and their system pretty much still stays off. But if the G12 locus is mutated, so pretty much if there is like any mutation in this particular region that is representative of colorectal cancer, then that off signal is removed, and then your output is by default turned on, and expression from the output gene turns on. We tested this natural DNA sensor designed in vitro using PCR products from blah blah blah, an archaic genome. This is donor DNA. Natural DNA biosensors with a random CRISPR spacer detected DNA sequences from both cell lines. Or E, right. And biosensors with the CRASP spacer accurately detected only DNA sequences from LS blah blah cells which contain the CRASP mutation, or E, demonstrating biosensor detection and discrimination of natural target DNA. And their system worked. Let's see. Any fraction of biosensor with random CRISPR spacer, D, or a spacer targeting wild type E. Okay. This is not super critical. Dissect. All that to say is like pretty much like their system is working as intended, as in they only get their on signal when there's that mutation that pretty much that colorectal cancer mutation. Whereas they sort of say here, demonstrating biosensor detection and discrimination of natural target DNA. The sensor, so wrapping up in their discussion, the sensor bacteria described here demonstrate that a living biosensor can detect specific mammalian DNA shed from colorectal cancer in vivo in the gut. But, it's, at least in this system, they moved out, they only tested up to the mouse gut with no sample preparation or processing. And that is pretty critical. And that's why they tested that. Because, this would hold very little advantage if, it required intensive sample processing. And it required, a lot of technical expertise. But no, they want to make this as simple as possible, with ideally with minimal to no sample processing or preparation required. They essentially want to go from straight stool sample to or they're kind of ideal system if it works as intended. It's you take human stool sample, and then maybe with some just cleaning up of the sample, or even no cleaning up of the sample, just maybe diluting it in a specific way and then placing it on a growth plate. And if you have growth, then that means that there are almost certainly colorectal cancer cells present in this person's gut. But if you have to go from a stool sample to multiple steps of processing or purification, then pretty much there's very little advantage, or advantage of this is very much reduced, as it just adds more steps to the process. Engineered donor cassettes are not required for catch biosensors to detect, discriminate, and report on target sequence. Although the final natural DNA biosensors will need an improved signal-to-background ratio to reliably detect sequence within whole genomic DNA, the homology arms and CRISPR spacers are modular, so this strategy could be readily adapted to detect and analyze arbitrary target sequences of interest. And so they're pretty much highlighting here that it doesn't need to be only for colorectal cancer. On paper, this should be adaptable to other systems or other types of cancers, other organs maybe, or in other locations of the body. Our technology is not yet ready for clinical application. This approach requires further development to ensure that future versions, at least those designed for gastrointestinal use, may be delivered orally and achieve sufficient luminal density to allow reliable detection by non-invasive samples, such as in stool or blood. As the technology advances toward clinical care, we'll also need to more critically evaluate the performance of catch compared to other relevant disease-specific tests, such as, in this case, colonoscopy and in vitro nucleic acid assay. Right. So again, this is all, you can pretty much think of this in the prototype stage of what they're doing is very cool, very ambitious, and requires a lot of technical expertise. Like I said, it's a multidisciplinary team of researchers and experts coming together to develop this, but it's still very much preliminary, and they're highlighting some of the future technical hurdles that they want to overcome. One of them is so when they were working with rodents, okay, in order to deliver their system or their engineered bacterial biosensor, they just inserted it through colonoscopy, pretty much through invasive means into the mouse. Okay, that is not ideal, or ideally you don't want to be doing that to humans, of hey, if we want to get our engineered biosensors into me, ideally we don't want to subject you to a colonoscopy. And so is this adaptable to, let's say, a pill form, or something that you can just take in a pill, or as they say, delivered orally? Um, and also, if it's delivered orally, know, it has to pretty much, go through your entire gastrointestinal tract, be digested at some point, or like the contents, like if it's, let's say, in a pill, and the bacteria, the engineered biosensor bacteria are inside of a pill. Well, it needs to like get out of that pill. It needs to get out of that pill and into, your large intestine, so that it can colonize it and like be there and do its detecting thing. And they're saying here, it's like, well, not only do we need to adapt it in, let's say, pill form, it needs to be delivered to our site of interest, which is large intestine, and then the bacteria actually need to be able to grow, as they're highlighting here, with technical jargon of achieve sufficient luminal density. That's just fancy terminology of like, hey, do they actually grow enough, and to grow to a sufficient density or amount that like we can actually like recover it. As in, like, if it literally turns out that, hey, you swallow a pill, and it just like literally only one engineered bacterial biosensor like makes it to your large intestine, that is absolutely useless. Just having only one bacterial biosensor present there is not going to do anything. Their system is not adapted to that, to just be able to detect one bacterial biosensor. They need to recover the bacterial biosensor and actually grow them, in this case, on a plate, grow them in something. And the likelihood that they'll be able to recover, that one single engineered biosensor is unlikely. So they need to be there in a certain amount, or as they say, in a certain density. And they're not yet sure whether they can actually do that. But that's something that they'll have to look at some point. Like, hey, not only is it there, but it's actually there in sufficient quantities that like we can actually use it, detect, or like recover and then detect. And then we also need to more critically evaluate the performance of catch, which that their detection system for biosensor compared to other relevant disease-specific tests. So how does it compare to conventional ways that we do screening now? And in the example that they're using is colonoscopy, since they're detecting colorectal cancer, and pretty much one of the more conventional methods, as highlighted earlier, for colorectal cancer detection or screening is through colonoscopy. And so, like, how does it actually stack up to that? Is it better, worse, the same, or even just like cost-benefit analysis of like, this system is useless? If it's like, it requires like $1 million to get it to one patient. Even if it proves like, better or marginally better than colonoscopy, well, if there's that cost barrier, there's not going to be any option of it. So like, how does it stack up to actual colonoscopy, or other ones that are used? Further bioengineering is required to limit the risk of biosensors escaping circuit-mediated cell death and to improve the efficiency of natural DNA detection, right? They're highlighting also another key thing, because they are engineering bacteria. And as mentioned, their ultimate end goal is like produce them in humans, or at least that's their ultimate ambition. Whether they actually get there is a different story. And so it's like, well, what are some of the concerns with engineering bacteria and administering it to humans? And these are very specifically engineered bacteria, and some of them are engineered with antibiotic resistance. And so it's like, do we want to do that? Want to be giving these bacteria to humans? Or like, is there a risk of like these bacteria getting out into nature? We arguably, we might be administering, hazardous materials, or potentially hazardous materials, hazardous to the environment, if they were to get out to humans. But they've engineered, it looks like they've engineered, or like they have some fail-safes engineered in the system that should hopefully prevent that. They don't actually know that it works for sure. As our technology progresses, careful analysis is essential to ensure patient safety, to minimize the risk of spreading antibiotic resistance, and to satisfy biocontainment concerns. Right. Because, if these are released in the stool, right, and coming from like a stool sample, and it's like, and if these bacteria are contained in, like, let's say the large intestine of the human, well, then there's a chance that they might get out, or they might not be contained much as we would want. We need to make sure at some point they are actually being biocontained. These necessary next steps are being actively pursued and are important as catch is applied to additional preclinical models and before it is trialed in humans. In vitro DNA analysis helps detect and manage important human diseases, including cancer and infection. However, in vitro sensing requires potentially invasive removal of samples, and many DNA diagnostics do not achieve clinically relevant sequence resolution, with more advanced techniques remaining too expensive for routine use in all settings. So they're mentioning some of the drawbacks or clear shortcomings of other conventional methods, or highlighting what should be some of the advantages of their system. Where, yeah, invasive. So, using the example of colorectal cancer, very often, not only is colonoscopy invasive, but also, as they're highlighting, if you want to, maybe detect cancerous cells or precancerous cells, well, you need to do a biopsy. There is some sampling, as opposed to here, the sample is coming from this, or like, the bacteria, the engineered bacteria are, taking up the sample for you, and it's being, expelled in the stool. Direct sampling of the gut in vivo may offer important advantages. The gastrointestinal tract contains marked deoxyribonuclease activity, or DNAs. This is just fancy terminology of, like, your gastrointestinal tract has the ability to degrade DNA, sort of analogously what I highlighted earlier with the CRISPR-Cas system, and the bacterial defense system, sort of like the bacterial immune system. It's adapted to degrade DNA, but specific types or specific DNA or genetic sequences. But so it's discriminatory. But in the case of our GI tract, it has the ability to degrade DNA, but it is not discriminatory. As in, pretty much, over time, degrade just about any DNA that is present. And that is a drawback, as they say, which limits the lifetime of free DNA in both rodents and humans, and may thus reduce the information content of downstream fecal samples. Right, because the question becomes, like, why do you need the bacterial biosensor to begin with? Because we're using the bacterial biosensor to pretty much detect genetic information, or cancerous genetic information. And so, if there's cancerous genetic information in the GI tract, well, then it should be there, when you go to the bathroom anyways. It should already be in the stool. But, as they're highlighting here, it's potentially not there, because essentially the longer it stays in the GI tract, greater the chances that it's going to be degraded. That's just sort of one of the native functions of your GI tract. Just as it pretty much digests and breaks down foods into macromolecules or molecules. No surprise, it will also break down genetic information. So it's like, yeah, if you have colorectal cancer cells that are releasing cancerous genetic information, it's just going to be destroyed by the GI tract, thus not detectable. And one of the purposes of the bacterial biosensor is to overcome that challenge. As they highlight, bacterial biosensors located in situ, that's just fancy terminology for, like, at the site, or at location in large intestine, could capture and preserve DNA shortly after its release before degradation by local DNases, pretty much local DNA degrading machinery. And that's pretty much one of the advantages of their system. Of like, hey, if their engineered bacterial biosensor is already there and it has the ability to uptake information, well, once the cancerous genetic information is inside the bacterial biosensor, it should be protected, or rather, it will be protected from DNA degradation. Essentially, going to preserve it, and then the bacterial biosensor will do its thing. So perhaps the most exciting aspect of catch, however, is that unlike in vitro diagnostics, once target DNA is captured, it could be coupled to direct and genotype complementary delivery of nanobodies, peptides, or other small molecules for the treatment of cancer or infection. Catch allows for the cellular detection of cell-free DNA, and thus may prove useful in future synthetic biology applications wherever and whenever DNA detection and analysis are important. So they're wrapping up, basically, hypothetically, one of the huge advantages of their system. Pretty much, this is focused on detection or screen. And like, hey, does our system have the ability to detect colorectal cancer? And the answer appears to be yes. But the overall advantage, potentially, of their system is not only detection, but also treatment. As in, they've highlighted, or they've tried to highlight, that their system is very modular. What that means, sort of casually, it's like the best thing that I can come up with, it's like a plug and play system. They're using very specific, parts here in their system, and they're engineering their bacterial biosensor in a very specific way. But there's no reason to think that it's limited to just what they're doing here. Certain other things can be introduced, and well, they even did it, because like this system here towards the end, where they're using pretty much a fairly different system, but the overall output is the same, already showing that it can be adapted to behave differently. And what they're saying is, not only can it be adapted for detection of, let's say, a disease or condition, or a cancer, or some other disease that requires DNA detection. You can also potentially couple it to treatment, and they highlight treatments such as nanobodies, peptides, or other small molecules, because again, what they're doing here is they're engineering bacteria, and in this case they're engineering bacteria to detect DNA, and they're doing it by a specific mechanism of if there is cancerous, if there's the presence of cancerous genetic information, and there will be an exchange of genetic information between the engineered bacteria, the biosensor, and that cancerous genetic information. And then the bacteria will gain a new function, and then it will be able to grow, in this case, the presence of antibiotic. But you can hypothetically, do more genetic manipulation, where you have a system that, if the bacteria, has that exchange of genetic information, and, it acquires that antibiotic resistance, you can design it such that, if they grow and you introduce, new genes and you design it appropriately, this is where the synthetic biologist comes in, of pretty much introducing a brand new part where you have two things going on at once, or multiple events of not only just the detection aspect, but also the treatment aspect of hey, if you're growing in the presence of kanamycin, well, that's great, but then also turn on this gene that makes the treatment, in this case, which could be a nanobody, that can totally be encoded in genetic information. Same thing with a peptide. Small molecule might be more challenging. Totally, nanobodies and peptides. Those are protein-based treatments, and proteins are derived from genetic information. Or rather, proteins are translated from genetic information. And so you can totally have it again, such that the system works where it's not only detecting, but also once it detects it, it produces the treatment. That might be in the form of a nanobody or a peptide. And that would be one of the true powerful advantages of their system. But again, if they get it fully up and running, as they highlight and emphasize, this is still very much in the experimental stage, and there's a lot of technical challenges still to overcome. But I would say this is a pretty powerful proof of principle, and there's a reason why this was published in Science and published recently. Although recency has nothing to do with it, but yeah, the fact that it's published in Science, even though the prestige of a journal doesn't mean much to me, the reality is there is prestige publishing is a thing. This wasn't published in some no-name journal that nobody has heard about. This is high impact journal publication, and there's a reason why this is not only is what they're doing technically challenging, even though I would say that, in my opinion, my non-expert opinion, because I'm not a synthetic biologist, but in my opinion, their system is like very elegant. But if anything, that's that's a testament to their expertise or their technical expertise. To design a system that works, and it seems to work robustly. And they scaled it up to fully, pretty much a full mouse model. But at the end of the day, still pretty elegant. So that's what I wanted to dissect about this paper. And do I actually have time to go through the other piece of research that I wanted to? Answer, yes. And I don't think I'm gonna be able to do justice to the paper. So I'm gonna probably put this on the back burner. Covering this took a little bit longer than I expected, but that's not a complaint. Since the more time that I can spend on a paper, better. So I'm probably gonna save this for tomorrow. I do plan to be back tomorrow, but in the meantime, I do have some shorter articles that I want to go through. That sort of touch upon what we were just covering. Well, maybe a glimpse into what the future holds. Where this system, well, not this, this system was, designed by humans, carried out by humans. This is synthetic biology, as highlighted here. But, we now have AI or large language models, and there's just more and more research coming out showing that the power of these large language models in a variety of fields, from medicine to whatever, reading, writing, whatever, publication, and also biology. And I do have some research that I'm preparing and going through in terms of AI and biochemistry and proteins. I just need to organize it and put it together. But large language models like GPT-4 have taken the world by storm thanks to their astonishing command of natural language. Yet the most significant long-term opportunity for large language models will entail an entirely different type of language of biology. Oh, Bob Marley, welcome. Thank you so much for passing by. Hope you're doing well. I imagine you're in Germany, or maybe have returned home to the UK. We just finished going through this engineered bacteria to detect your DNA. And now we're going over this. And there was a paper that I'm probably going to cover soon that actually made me think specifically sort of you, or my resident economist. And I might start calling economists, pro-brained, because there was an interesting paper published about how, crows, the birds, they might have underestimated their ability, their cognitive ability, and they might be carrying out some pretty complicated calculations in their behavior. Where was it? Yes. One striking theme has emerged from the long march of research progress across biochemistry, molecular biology, and genetics over the past century. It turns out that biology is a decipherable, programmable, and in some ways even digital system. DNA encodes the complete genetic instructions for every living organism on Earth using just 4 variables: A, C, T, and G, or adenine, cytosine, guanine, and thymine. Compare this to modern computing systems, which use 2 variables, 0 and 1, to encode all the world's digital electronic information. One system is binary and the other is quaternary, but the two have a surprising amount of conceptual overlap. Both systems can properly be thought of as digital. To take another example, every protein in every living being consists of and is defined by a one-dimensional string of amino acids linked together in a particular order. Proteins range from a few dozen to several thousand amino acids in length, with 20 different amino acids to choose from. Right. Where I've used this example multiple times previously, but I think of amino acids, of which there are, at least generally speaking, in humans, or mammals, or pretty much like most living organisms. There are typically 20 canonical amino acids. I think of them as Lego blocks, where proteins are composed of amino acids, and you can, just as you can have, something simple, like a house made of Legos, or even smaller. You can have something that's super complex, as just about anything made of Lego. Of like whatever, the Millennium Falcon, or like, an actual full-sized house made of Lego. But yeah, pretty much these 20 amino acids, the quote-unquote Lego blocks, can be assembled into so many different things, to so many, almost an infinite variety of protein, ranging from, whatever, small, minuscule protein to something, to a large, huge macromolecular protein complex, where it's like, yeah, a few dozen to several thousand. This too represents an eminently computable system, one that language models are well suited to learn. As DeepMind CEO co-founder Demi Hassabis put it, at its most fundamental level, I think biology can be thought of as an information processing system, albeit an extraordinarily complex and dynamic one. Just as mathematics turn out to be the right description language for physics, biology may turn out to be the perfect type of regime for the application of AI. Large language models are at their most powerful when they can feast on vast volumes of signal-rich data, inferring latent patterns, and deep structure that go well beyond the capacity of any human to absorb. They can use this intricate understanding of the subject matter to generate novel, breathtakingly sophisticated output. Have you seen the research? I have not, but if you want to share it, either here or in the Discord, by all means, feel free. I know you're already in the Discord, just for anybody else that's in. But I'd be totally interested in that research. By ingesting all of the text on the internet, for instance, tools like ChatGPT have learned to converse with thoughtfulness and nuance on any imaginable topic. By ingesting billions of images, text-to-image models like Midjourney have learned to produce creative original imagery on demand. Pointing large language models at biological data, enabling them to learn the language of life, will unlock possibilities that will make natural language and images seem almost trivial by comparison. What concretely will this look like? In the near term, the most compelling opportunity to apply large language models in life sciences is to design novel proteins. Well, I trust your judgment. And also, I'm not perfect, but I think I'm decent at vetting a preprint. Unless it's like, an LK99 preprint. Then I'm useless there. Proteins are at the center of life itself. Right? And I think this is, an often, at least to the general public, an often overlooked thing. Everybody knows about DNA and maybe RNA. But proteins, as nicely puts it, are at the center of life itself. I like to think of, or at least the best analogy that I can come up with when teaching about proteins to other people, or like, when talking about biochemistry to those who are not in biochemistry or like not in the sciences. It's like, if biology is like a movie, okay, DNA is the script, or like, whatever, the stage directions. But yeah, I think the best thing is script, or like the screenplay. But when you actually watch a movie, what you see are the actors. What actually plays out are the actors, and I would say the proteins are the actors. As in, the proteins are the ones doing the thing in a biological system. But yeah, proteins are at the center of life itself. As prominent biologist Arthur Lessig put it, in the drama of life at a molecular scale, proteins are where the action is. Oh, I haven't pre-read this, but kind of stealing my analogy. But I would say not only just where the act, but yeah, they are the actors on screen. What you actually see. So that's the most interesting aspect of a movie. What you're watching. Not the script, not the screenplay, not the director. It's what plays out on screen. Proteins are involved in virtually every important activity that happens inside every living thing: digesting food, contracting muscles, moving oxygen throughout the body, attacking foreign viruses. Your hormones are made out of proteins, so is your hair. Proteins are so important because they are so versatile. They are able to undertake a vast array of different structures and functions, far more than any other type of biomolecule. This incredible versatility is a direct consequence of how proteins are built. As mentioned above, every protein consists of a string of building blocks known as amino acids, strung together in a particular order. Based on this one-dimensional amino acid sequence, proteins fold into complex three-dimensional shapes that enable them to carry out their biological functions. A protein's shape relates closely to its function. Take one example. Antibody proteins fold into shapes that enable them to precisely identify and target foreign bodies, like a key fitting into a lock. As another example, enzymes, proteins that speed up biochemical reactions, are specifically shaped to bind with particular molecules and thus catalyze particular reactions. Understanding the shapes that proteins fold into is thus essential to understanding how organisms function and ultimately how life itself works. Yeah, that is a totally valid question. So I'm not a follower of Forbes, and I generally don't read much. I read the occasional article, and maybe I read an article from them like once every 2 months, like a few times a year. But this article came to my attention through a reputable source, as in like someone pretty much a credible person on social media that I follow shared this article, and so it caught my eye. But it's totally possible that it's an absolute miss. But so far, what they're saying here is pretty milquetoast. Not really saying, my opinion, not really anything controversial. At least not yet. And thank you so much for the preprint. Speaking of shoe, I did do some running today. And I'm sort of like leaning towards, hopefully at some point, migrating towards that minimal shoe footwear. Stanford University. Stanford University in my mind. When I covered that resignation of the president of Stanford, a huge downgrade. Although I didn't think much of Stanford University. Well, I don't think much of just about any university, regardless of name. Or rather, fancy names like Stanford and Harvard don't really mean much. This is all just branding. Although I'm joking. I'm sure this is like overall story. But I do kind of have like a little bit of a mild bias against I ran for a good 55 minutes at a pretty slow pace, actually. Like, I ran for about 7.5, 8 kilometers. So I was running around 8 kilometers per hour, so not super fast. It was really like a casual, low impact run. But it felt good. And I still feel energized. Where was I? Yes. Determining a protein's three-dimensional structure based solely on its one-dimensional amino acid sequence has stood as a grand challenge in the field of biology for over half a century. Referred to as the protein folding problem, yeah, absolutely. It has stumped generations of scientists. One commentator in 2007 described the protein folding problem as one of the most important yet unsolved issues of modern science. Right. And I'll probably have to cover this on a future stream. This protein folding, even though I've covered, let's say protein structure on previous streams. I've covered it a little bit in depthly, but maybe not as in depthly as I would have liked, because yeah, until I get that tablet, which I'm pretty much committed to buying soon, until I can actually like draw things out, at least by hand, I'm very much limited in sort of like how I can translate issues and like protein and the protein folding issue and protein structure. But like, yeah, this is absolutely a thing. In late 2020, in a watershed moment in both biology and computing, an AI system called AlphaFold produced a solution to the protein folding problem. Built by Alphabet's DeepMind, AlphaFold correctly predicted proteins' three-dimensional shapes within the width of about one atom, far outperforming any other method that humans had ever devised. Yes, this is true. I think I even covered that publication. I think I even have. I definitely have. This is AlphaFold 2. That's not the original. I totally have other AlphaFold. I just need to. Guess they're not. This library. Although they should be. Oh well, whatever. I'll dig that up. But yes, so far what they're saying is true. It is hard to overstate AlphaFold's significance. Longtime protein folding expert John Moltz summed it up well. This is the first time a serious scientific problem has been solved by AI. Yet when it comes to AI and proteins, AlphaFold was just the beginning. AlphaFold was not built using large language models. It relies on an older bioinformatics construct called multiple sequence alignment. So yeah, I haven't used AlphaFold, but this multiple sequence alignment I have used. I mean, this is my research. I'm like, yeah, part of my research, like solving protein structure. This is our attempts at solving a protein structure, but it wasn't actually solved because this is a computer model. And I use multiple sequence alignment. Well, when we say multiple sequence alignment, we're pretty much generally sort of loosely referring to this. This is like the sequence of like a bunch of proteins, and we're using multiple of them to like, you know, pretty much trying to extract what we think this new protein looks like. Pretty much, we're using a known to solve an unknown. Of like, hey, these proteins have been solved, okay, and we know their structures. And this new one, we think it has an evolutionary history, or rather, its sequence of amino acids seems to match closely, or at least, you know, a certain threshold of like what we call sequence identity. Then hey, maybe this new protein looks like the other ones. They at least share some sequence similarities or identities. But like, you know, the structure that we generated here, like it's a model, a computational model. It is a prediction. We didn't actually solve it in reality. This was not uploaded to the protein database. Because as they highlight here, solving an actual structure of a protein, generally pretty large technical challenge. But yeah, multiple sequence alignment can be powerful, as AlphaFold made clear, but it has limitations. For one, it is slow and compute intensive because it needs to reference many different protein sequences in order to determine any one protein structure. More importantly, because multiple sequence alignment requires the existence of numerous evolutionarily and structurally similar proteins in order to reason about a new protein sequence, it is of limited use for so-called orphan proteins, proteins with few or no close analogs. Such orphan proteins represent roughly 20% of all known protein sequences. Right. That's why that's one of the challenges, or that would have been one of the challenges that I encountered, because, you know, I used a known in an attempt to solve or predict an unknown. But if there was no known, as in if these other structures weren't solved, then like, yeah, I'd have nothing to go by. I would literally have nothing to solve an unknown. And that totally happens, where sometimes we come across proteins where we look at them and they seem to be fairly, they're either fairly unique, as in at least we can't, it's difficult to trace their evolutionary history, or like what other proteins that they're related to, that they might share an evolutionary history with, or we just haven't, you know, solved other structures that share an evolutionary history with that new particular protein that we're trying to solve. Recently, researchers have begun to explore an intriguing alternative approach, using large language models rather than multiple sequence alignment to predict protein structures. Protein language models, large language models trained not on English words but rather on protein sequences, have demonstrated an astonishing ability to intuit the complex patterns and interrelationships between protein sequence, structure, and function. Say how changing certain amino acids in certain parts of a protein sequence will affect the shape that the protein folds into. Protein language models are able to, if you will, learn the grammar or linguistics of proteins. The idea of a protein language model dates back to the 2019 UniRep work out of George Church's lab at Harvard. So UniRep used LSTMs rather than, but yeah, this George Church person, fairly infamous. This person on the right here, fairly infamous in the world of molecular biology. In late, and whatever. no surprise, works at Harvard. In late 2022, Meta debuted ESM2 and ESM Fold, although I think these have since been abandoned, I think. Yeah. I remember coming across so it looks like Meta or Facebook was trying to advance the scientific endeavor, and it looks like they were trying to use AI to solve protein folding, but it seems like they have since ended it, which is unfortunate. But yeah, one of the largest and most sophisticated protein language models published to date, weighing in at 15 billion parameters. ESM2 is the large language model itself. ESM-Fold is its associated structure prediction tool. ESM2 or ESM-Fold is about as accurate as AlphaFold at predicting proteins' three-dimensional structures. And also we have, since moved on to AlphaFold 2. When was it? Wasn't this published recently? Yeah. Like a little bit, almost exactly a month ago. And they're talking about AlphaFold. But we totally have AlphaFold 2. I believe an upgrade, pretty large upgrade over AlphaFold. Maybe that's, as you say, Bob Marley, a miss. But yeah. But unlike AlphaFold, it is able to generate a structure based on a single protein sequence without requiring any structural information as input. As a result, it is up to 60 times faster than AlphaFold. When researchers are looking to screen millions of protein sequences at once in a protein engineering workflow, this speed advantage makes a huge difference. ESM-Fold can also produce more accurate structure predictions than AlphaFold for orphan proteins that lack evolutionarily similar analogs. Language models' ability to develop a generalized understanding of the latent space of proteins opens up exciting possibilities in protein science. But an even more powerful conceptual advance has taken place in the years since AlphaFold. In short, these protein models can be inverted. Rather than predicting a protein structure based on its sequence, models like ESM2 can be reversed and used to generate totally novel protein sequences that do not exist in nature based on desired properties. Right. So it's not only powerful in predicting what exists in nature, what presumably exists in nature, but it also might have power and utility in predicting new things or new functions. Pretty much in the domain of protein engineering. And I totally have, well, I think so I think this paper touches upon that. I believe this paper, if I remember correctly, they're using AlphaFold 2 for the purposes of engineering. But I totally have other research that I'm trying to prepare that touches exactly on this. This is one of them, sort of in that vein. Although it's not literally in the protein engineering, but it is sort of a prediction. And I think it's actually is using this ESM from Meta. But I haven't gone through this yet. But regardless, totally has potentially huge implications for protein engineering. All the proteins that exist in the world today represent but an infinitesimally tiny fraction of all the proteins that could theoretically exist. Herein lies the opportunity. To give some rough numbers, the total set of proteins that exist in the human body, the so-called human proteome, is estimated to number somewhere between 80 and 400,000 proteins. 400,000. Accurate. If I remember correctly. Meanwhile, the number of proteins that could theoretically exist is in the neighborhood of, 10 to the power 1300, or like a lot. An unfathomably large number, many times greater than the number of atoms in the universe. To be clear, not all of these 10 to the power 1300 possible amino acid combinations would result in biologically viable proteins. Far from it. Some subset would, well, and that is actually one of the puzzles, or was one of the puzzles of protein. Because again, well, it's called, so, you know, it's called AlphaFold for a reason. Going back to AlphaFold, I keep placing. But yeah, it's called AlphaFold because proteins fold. And like it was AlphaFold. And yeah, because proteins fold, where you have your amino acid sequence, you know, you can think of it as like a linear or like string-like sequence of amino acids, but they fold into very intricate conformations. They should, So like, I have some pictures here of these very intricate conformations, although this is sort of well, like a cartoon-ish depiction, but it still, I think, gets the message across that, you know, you're going from they have this nice figure here, sort of in the blue, so they're contrasting the blue lettering, okay, that's the amino acid sequence. Each letter is an amino acid. So this M, that stands for methionine, this H, histidine, threonine, glycine, arginine, well, you get the point. But like, so you have, you know, your amino acid sequence, but in the yellow cartoon above the blue lettering, that is the structure. That's what it folds into. And at least for those sequences, they're saying here in this sequence, it folds into an alpha helix, where it has this helical structure. And one of the puzzles that sort of they alluded to in the Forbes article, it's like, you know, there's essentially given that, you know, you just have this string of amino acid sequence, it can fold into pretty much in terms of possibilities. It has like almost infinite possibilities of what it can fold into. But it but it turns out to be the case that they're folding and adopting these conformations like in a very specific way. Like it looks like it's guided. And like that is like, you can pretty much like adopt, like, you know, you're going from like infinity, but you're always like zeroing in on like this one particular protein conformation. And so that, like, at a conceptual level, that was like at one point one of the challenging things about proteins, kind of like the protein folding problem, of like, hey, how come they're not just adopting, you know, random conformations? You get this sequence, and then it's like, well, this sequence always seems to ends up in this particular structure. So what's going on there? You know, that has since like been solved of like why proteins fold in these specific ways that I'll probably cover on a future stream. I'll probably have to wait till I get my tablet for that to start drawing things out. But yeah, over many millions of years, the meandering process of evolution has stumbled upon tens or hundreds of thousands of these viable combinations. This is merely the tip of the iceberg. In the words of Molly Gibson, co-founder of leading protein AI startup Generate Biomedicines, the amount of sequence space that nature has sampled through the history of life would equate to almost just a drop of water in all of Earth's oceans. An opportunity exists for us to improve upon nature. After all, as powerful of a force as it is, evolution by natural selection is not all-seeing. It does not plan ahead. It does not reason or optimize in top-down fashion. It unfolds randomly and opportunistically, propagating combinations that happen to work. Using AI, we can, for the first time, systematically and comprehensively explore the vast uncharted realms of protein space in order to design proteins unlike anything that has ever existed in nature, purpose-built for our medical and commercial needs. So actually, I think this overlaps nicely with the paper that I covered, because this is, we're talking about protein engineering, and this is biology and engineering. We just call it synthetic biology, like engineering organisms for our purposes. And as highlighted here, this on paper, this is a potentially powerful purpose if it actually, gets up to scale and fully off the ground, of like detecting cancer or detecting maybe a whole host of other diseases and conditions. Not only just detection, but as they say towards the end, also potentially treatment, or like coupling treatment with detection of like a bacterial biosensor that, hey, if it detects the cancer, well, it just also turns on the gene for the treatment of that cancer. And maybe one of the advantages is it's like, right at the site of the location of the cancer, like, the disease tissue or organ or whatever. Where, we are pretty much like hijacking things in nature, and we're just like, tinkering around of like, the novel aspect isn't, the genetic sequence or anything like that. It's like we are pretty much repurposing bacteria, or like we're engineering bacteria to suit our human needs, or at least with the ambition to suit our human needs. And the same thing holds true for protein, where proteins are currently, there have been engineered proteins, okay? But these have, let's just say, they haven't been high resolution engineering of proteins yet. We've come up with, let's say better insulin, and, we've come up with, engineer, well, maybe not engineer, but, designed antibodies. But like, we're not quite at the place of like, hey, this is what our prototype protein looks like on paper. Here's its amino acid sequence as predicted by, let's say, AI, and then now let's go make it or something. We're definitely not there yet. But yeah, using AI, we can, I already read that. We will be able to design new protein therapeutics to address the full gamut of human illness, from cancer to autoimmune diseases, from diabetes to neurodegenerative disorders. Looking beyond medicine, we will be able to create new classes of proteins with transformative applications in agriculture, industrials, material science, environmental remediation, and beyond. And I sort of highlighted this a little bit. Again, I covered this a couple months ago, or maybe longer. But like, yeah, this antifreeze protein, when I was talking about proteins and protein structure, and like this protein had, not only just a cool, well, at least me cool, structure, this like 4 helix structure, but it also had, a pretty cool function. This is an antifreeze protein. And there were multiple cool things about this. I actually covered this paper in one of my graduate courses, and also published in Science, and published in Science for a reason, because like super cool. And there are multiple, let's say, novel or new things as a result of. But like, yeah, this was a protein found in nature. It's an antifreeze protein, but it was found in organisms. I think in this instance, fish. Could be wrong, but some sort of like fish that lives in like really cold waters, like approaching freezing waters, or I think even like below freezing waters. And like whatever, if it's living in these environments, well, the fish itself doesn't want to freeze, so it needs to have some sort of ability to like break up ice crystals or prevent the formation of ice crystals. And it has resulted in the evolution of this protein that has the ability to bind to ice, and prevent the formation of ice, or like actually break down ice. And with appropriate engineering, or like appropriate design and engineering, this hypothetically can be adapted to human purposes, for sure, for multiple human purposes. Some early efforts to use deep learning for de novo protein design have not made use of large language models. One prominent example is protein MPNN, which came out of David Baker's world-renowned lab at the University of Washington. Rather than using large language models, the protein MPNN architecture relies heavily on protein structure data in order to generate novel proteins. The Baker lab more recently published RF diffusion, a more advanced and generalized protein design model. As its name suggests, RF diffusion is built using models, the same AI technique that powers text-to-image models like Midjourney and Stable Diffusion. RF diffusion can generate novel, customizable protein backbones, that is, proteins, overall structural scaffoldings, onto which sequences can then be layered. How long? Article. Okay. I was like looking at the scroll bar. Oh, a lot of scrolling. But no, most of it's garbage. Where was I? One of the key forces behind the dramatic recent progress in artificial intelligence is so-called scaling laws. The fact that almost unbelievable improvements in performance result from continued large language model parameter count, training data, and compute. At each order of magnitude increase in scale, language models have demonstrated remarkable, unexpected, emergent new capabilities that transcend what was possible at smaller scale. It is OpenAI's commitment to the principle of scaling, more than anything else, that has catapulted the organization to the forefront of the field of artificial intelligence and not. As they move from GPT-2 to GPT-3 to 4 and beyond, OpenAI has built larger models, deployed more compute, and trained on larger datasets than any other group in the world, unlocking stunning and unprecedented AI capabilities. Our scaling law is relevant in the realm of proteins, thanks to scientific breakthroughs that have made gene sequencing vastly cheaper and more accessible over the past 2 decades. The amount of DNA, and thus protein sequence data, available to train AI models is growing exponentially, far outpacing protein structure data. Protein sequence data can be tokenized for all intents and purposes, treated as textual data. After all, it consists of linear strings of amino acids in a certain order, like words in a sentence. I don't think that's the best analogy, but I understand why. But like, yeah, no, it's just that sequence, linear sequence of amino acids, highlighted here from this paper in the blue text. But this linear sequence, folds into intricate three-dimensional conformations. Large language models can be trained solely on protein sequences to develop a nuanced understanding of protein structure and biology. This domain is thus ripe for massive scaling efforts powered by large language models, efforts that may result in astonishing emergent insights and capabilities in protein. The first work to use transformer-based large language models to design de novo proteins was ProGen, published by Salesforce Research in 2020. The original ProGen model was 1.2 billion parameters. Ali Madani, the lead researcher on ProGen, has since founded a startup named Profluent Bio to advance and commercialize the state-of-the-art in large language model-driven protein design. While he pioneered the use of large language models for protein design, Madani is also clear-eyed about the fact that by themselves, off-the-shelf language models trained on raw protein sequences are not the most powerful way to tackle this challenge. Incorporating structural and functional data is essential. The greatest advances in protein design will be at the intersection of careful data curation from diverse sources and versatile modeling that can flexibly learn from that data, Downey said. This entails making use of all high-signal data at our disposal, including protein structures and functional information derived from the laboratory. Another intriguing early-stage startup applying large language models to design novel protein therapeutics is Nabla Bio, spun out of George Church's lab at Harvard and led by the team behind UniRep. Nabla is focused specifically on antibodies. Given that 60% of all protein therapeutics today are antibodies, and that the two highest selling drugs in the world are antibody therapeutics, it is hardly a surprising choice. And I think I've covered this before, sort of, like, yeah. Antibodies as therapeutics has just like really taken off, and that pretty much like a lot of the top selling drugs, or like the most profitable drugs, are now antibody based. And I think like for the past decade or so, like, number one has just like this one particular one, Humira. Well, pretty much, that particular antibody therapeutic, like, put this pharmaceutical company Abbott on the map. And that's why they take very aggressive action to protect their intellectual property. And they just not gonna get into it, but they basically try to legally protect their intellectual property, the antibody, as aggressively as possible. Nabla has decided not to develop its own therapeutics, but rather to offer its cutting-edge technology to biopharma partners as a tool to help them develop their own drugs. Expect to see much more startup activity in this area in the months and years ahead as the world wakes up to the fact that protein design represents a massive and still underexplored field to which to apply large language models' seemingly magical capabilities. In her acceptance speech for the 2018 Nobel Prize in Chemistry, Frances Arnold said, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it. The code of life is a symphony, guiding intricate and beautiful parts performed by an untold number of players and instruments. Maybe we can cut and paste pieces from nature's compositions, but we do not know how to write the bars for a single enzymic passage." As recently as 5 years ago, this was true. But AI may give us the ability for the first time in the history of life to actually compose entirely new proteins and their associated genetic code from scratch. Purpose-built for our needs, it is an awe-inspiring possibility. These novel proteins will serve as therapeutics for a wide range of human illnesses, from infectious diseases to cancer. They will help make gene editing a reality. That might be vicious. Maybe. I think they'll certainly help, but I don't know if that alone is going to make gene editing a reality. They will transform materials science. They will improve agricultural yields. They will neutralize pollutants in the environment, and so much more that we cannot yet even imagine. Yeah, the potential is there. I don't know what timeline they're thinking about, but yeah, I think this. Not necessarily an exaggeration, but maybe too much too soon. I don't think this is coming in the next few years, but I could be totally wrong. The field of AI-powered and especially large language model-powered protein design is still nascent and unproven. Meaningful scientific engineering, clinical, and business obstacles remain. Bringing these new therapeutics and products to market will take years. Yet over the long run, few market applications of AI hold greater promise. In future articles, we will delve deeper into large language models for protein design, including exploring the most compelling commercial applications for the technology, as well as the complicated relationship between computational outcomes and real-world wet lab experiments. Let's end by zooming out. De novo protein design is not the only exciting opportunity for large language models in the life sciences. Language models can be used to generate other classes of biomolecules, notably nucleic acids. A buzzy startup named Inceptive, for example, is applying large language models to generate novel RNA therapeutics. Other groups have even broader aspirations, aiming to build generalized foundation models for biology that can fuse diverse data types spanning genomics, protein sequences, cellular structures, epigenetic states, cell images, mass spectrometry, spatial transcriptomics, and beyond. The ultimate goal is to move beyond modeling an individual molecule like a protein, to modeling proteins' interactions with other molecules, then to modeling whole cells, then tissues, then organs, and eventually entire organisms. I mean, true, this is also like a very ambitious endeavor. The idea of building an artificial intelligence system that can understand and design every intricate detail of a complex biological system is mind-boggling. In time, this will be within our grasp. Yeah, sure, on a long enough timeline. Probably, but I'm curious as to what timeline they're thinking. And like, yeah, who is... I guess they don't give bio. But yeah, I'm curious on their background. Not that anything that they're saying is like bad or necessarily inaccurate. But yeah, I think a little bit over exaggerating. The 20th century was defined by fundamental advances in physics, from Albert Einstein's theory of relativity to the discovery of quantum mechanics, from the nuclear bomb to the transistor. As many modern observers have noted, the 21st century is shaping up to be the century of biology. Artificial intelligence and large language models will play a central role in unlocking biology's secrets and unleashing its possibilities in the decades ahead. Buckle up. Oh, okay. Robert Toes is a venture capitalist at Radical Ventures. Okay. Then, I don't know for sure, but I suspect they don't have much scientific training. Venture capital firm. Then yeah, I would take, again, nothing that they're saying is necessarily inaccurate or wrong. I think there's a little bit of a grain of salt associated with the article. But overall, I think a pretty good overview in terms of like, yeah, there is a lot of power and potential there. But I think there's still a lot of, just as there's a lot of power and potential, but, you know, just like this is actual research, you know, in development, potentially in the pipeline, or, you know, hopefully on the way to human applications. But as they highlight here, still a lot of technical challenges that they need to overcome. And so it's like still quite a bit of time. And I think the same thing holds true of like, yeah, a lot of power and potential, but still, you know, a lot of technical challenges to overcome. That, you know, while AI certainly offers a lot of promise, but I don't think AI is going to solve at least all things that are highlighted. But could be wrong. But still, overall, pretty decent art. And yeah, I don't think I have time. Yeah, do not have time to cover this. Or actually, I definitely did not cover all of the content that I wanted to cover, but that is totally fine. Just means that this I'm going to cover tomorrow. Again, assuming my sleep holds up, which it should. But yeah, I'm gonna end the stream here. Right. But I totally had other content that I wanted to cover. Some other articles. Also going back to, you know, scientific fraud as well. I also wanted to cover some of the science memes in the Discord. But that's also gonna have to wait until covering this article or this research took way longer than I expected. That is absolutely not a... Thoroughly enjoyed dissecting this article. But yeah, gonna end the stream here and pretty much gonna pick up where I left off tomorrow. Let's see if I can find someone to raid. Lewd Hentai Girl, thank you so much for passing by. Hope you're doing well. It's nice to see you. Astro Canuck is thing. Yes, You did. That's okay, because we're gonna raid into a cool streamer. But yeah, I covered this, which is a cool paper about engineered bacteria to detect tumor DNA. But I'll be back tomorrow covering this and probably also some other cool research. And yeah, I definitely didn't get to cover all the content that I wanted to cover. That's totally not a problem. Get to cover it tomorrow. I'm gonna set up the raid to Astro Canuck, who's a great science and technology and arts and crafts streamer. They also do Lego build streams. But yeah, they're an astrophotography streamer and they're Canadian. Refrigerator. Actually, yeah, totally. I have it. I have. Well, whoopsies. There's this antifreeze protein. This research about an antifreeze protein. I had this already up, not obviously planned. But like, yeah, research about might be able to solve at some point. Your frozen or refrigerated burger. Where, you know, I'm sure there are engineering efforts to adapt or use this protein for human purposes, like thawing or defrosting or preventing the formation of ice crystals or ice formation. Where maybe at some, you know, we de-ice planes now. I don't know what they use, but maybe at some point it will be an engineered antifreeze protein that will almost certainly function much better than what we use now. Oh, but not frozen. There are totally I'm thinking you can totally cook a burger with some engineered protein. I'm just going through the filing cabinet in my brain, but I'm coming up with a blank. But still, it's doable. But yeah, I want to thank everyone that passed by. Lewd Hentai Girl, also appreciate the username. Thank you so much for passing by. Thank you, Doffy. Thank you, Bob Marley, and CSGO April. Thank you, Taller, for the raid. Super appreciate it. If you're not following them, you should be. And also thank you, my weird uncle, for the follow. HG Lewd. Okay, I'll try to keep that in mind. Can't make any promises. But yeah, you might have to remind me. Thank you, Boy Meets Mini, and also shout out to you. And CFC Hard R, thank you so much for passing by. I will be back, or rather, I should be back tomorrow, picking up where we left off. There's still a bunch of content that I didn't get to cover today, but I'll cover tomorrow, and also probably some new content if I have time for it. And also checking out the science meme in the Discord. That again, I want to cover today to get a chance to. Yeah, if anyone wants to join the Discord, feel free to either share research or ask questions or recommend a topic that you want me to cover. And yeah, we're gonna raid into Astro Canuck. If you're not following them, you should be, for multiple reasons. One, because they have a great stream design, super interactive. Two, because, you know, they're a science and technology streamer, astrophotography. And three, most importantly, they're Canadian. Although I believe they're from Ontario, so, you know, pseudo-Canadian, because I'm from Quebec, but still Canadian nonetheless. But yeah, thank you everyone. Super appreciated for anyone that passed by, and for the follows, and the raid. And until next time, take care, be well, be kind.
You're on Twitch.